Please be seated. Glory be to God. The Bible says, happy is the man that findeth wisdom, and the man that getteth understanding. A pleasant good morning to the members on the dais and off the dais, the enthusiastic participants through a virtual mode on this inaugural function of International Conference on Biological Challenges, BICPAC' 2002, sponsored by Department of Biotechnology and Council of Scientific and Industrial Research, Ministry of Science and Technology, Government of India. Before we begin this inaugural function, we should invite our Lord to get his guidance and blessing. Let us all rise up to invoke God's blessing. We will have an opening prayer. Now I request Dr. M.G. Daniel Andrew, Assistant Professor, to lead us with his opening prayer.
Our most gracious and loving Heavenly Father, King of the universe and Bishop of our souls, we thank you for this wonderful new day and for this conference on pandemic challenges and biological innovations and computer explorations. We thank you for your grace and your sustenance in keeping us alive, and for giving us vigor and breath in our nostrils. We thank you for the special invited guests for this conference and for this inauguration in particular. We thank you for the presence of Dr. M. Krishnan, the Vice Chancellor of Central University of Tamil Nadu, Tiruvallur, and for the other dignitaries whom you have brought safely in our midst. We pray, Lord, for your grace and your mercy on this conference, for all the plan that we have made to be executed properly. And we pray that there will be an explosion of knowledge coming out of this conference, that all the attendees and the participants will benefit richly, and that this will sow the seeds of knowledge and perpetuate this knowledge for decades to come. We thank you and we praise you for this great opportunity for our college. We give you all the praise and the glory in the precious name of our Lord and Savior Jesus Christ, we pray. Amen.
Thank you, sir. Kindly remain standing. Prayer. Prayer should not be regarded as a duty which must be performed, but rather as a privilege to be enjoyed, a rare delight that is always revealing some new beauty. Now, I invite our department choir to sing a prayer song, followed by State Anthem of Tamil Nadu, Tamil Thai Valthu, and college song. Anthem of Tamil Nadu, Tamil Thai Valthu. This light of happiness light up our BICPAC 2002 International Virtual Conference, which is going to be the grand event and will be a great success by God's mercy and grace. Thank you, dignitaries. A genuine leader is not a searcher for concerns, but a molder of concerns. Outstanding is how she always comes through with loyal, sincere, and full of good cheer, untiring in her efforts throughout the year, trustworthy in every aspect. She is very much eager to reach her every goal, effective in the way that she fulfills her role. Ready with a smile like a shining star, I invite Dr. Jasmine, Associate Professor, UG Head, and co-convener of this BICPAC 2002 to propose welcome address.
Thank you. A pleasant good morning to one and all gathered here. This is the day that the Lord has made. We will rejoice and be glad in it. Yes, indeed, we are overwhelmed with joy and we praise God who has enabled us to host this international virtual conference funded by DBT and CSIR. Welcoming is not just inviting people, but it is the warm gesture from our hearts. On this proud occasion, I'm extremely happy to welcome our eminent chief guest, Professor Dr. M. Krishnan, Honorable Vice Chancellor, Central University of Tamil Nadu, Thiruvarur, for inaugurating this international conference. We are indeed privileged to have such a person who has truly set a benchmark for his multi-hyphenated achievements. The presence of our Vice Chancellor here amidst us reveals his passion towards biology and his love towards the student community. The audience will be spellbound to know his achievements, which would be shortly disclosed. Your presence is a motivation for us all, sir. Thank you, sir, for your presence amidst your busy schedule. Welcome you, sir. I'm happy to welcome our beloved and dynamic principal, Dr. D. Paul Dayabaran, who has taken our college to great heights through his tremendous contribution. He has always been a support to the growth of our department. Welcome you, sir. I extend a warm welcome to our Vice Principal Aided, Professor Alakappa Moses, who works towards the success of our college and encourages and guides us in all our endeavors. Welcome you, sir. It is my pleasure to welcome our bursar, Dr. Nyan Raj, who always supports our department by graciously granting financial support whenever needed. Welcome you, sir. I'm very happy to welcome our beloved head of the department, Dr. Violet Dayabaran, who has joined online. She is an epitome of compassion who not only shows the way, but also goes the way. We are fortunate to work under her leadership. Welcome you, ma'am. I do extend a hearty welcome to our renowned, eminent speakers and delegates from all over the globe. I'm grateful to them for having accepted our invitation to share our expertise in this conference. I bid a very warm welcome to all the reputed judges who have consented to spare their valuable time to judge both the oral and the poster presentations. I also extend my welcome to deans, associate deans, heads, coordinators, and faculty from other departments who have graced this occasion with their presence. With great happiness, I welcome all the participants and faculty from other institutes who are connected online for this inaugural function. We are encouraged by their overwhelming response. At this moment, I recognize the presence of Dr. Ravi and Dr. Arvind, orthopedic surgeon from Kaveri Medical Hospital. And thank you, sir, and welcome you, sir. I am indebted to the organizing secretaries and my colleagues whose persistent effort and support has made this conference a reality. Welcome you all. I also welcome the non-teaching faculty members of our department. I deem it a pleasure to welcome all the enthusiastic students and office bearers who have stood up to the occasion by shouldering the various responsibilities rendered to them. Welcome, dear students. I'm happy to welcome the technical staff whose work behind the screen is appreciable. I once again welcome one and all to this 2 days international virtual conference on biological innovations and computational exploration for pandemic challenges, which is the need of the hour. Welcome one and all. Thank you.
Thank you, ma'am, for your hospitable address. Now, I request our principal, Dr. D. Pal Dayabaran, to honor our chief guest, Professor Dr. M. Krishnan, Honorable Vice Chancellor, Central University of Tamil Nadu, Thiruvarur. By adoring with a shawl, memento. As a token of love, on behalf of Department of Biotechnology and Bioinformatics, organizing committee members of this BICPAC' 2002. Thank you, dignitaries.
Now I invite Mr. Dinesh Raja, Organizing Secretary of BICPAC'22, to honor our eminent principal, Dr. T. Paul Dayabaran. Thank you, sir. I request Mrs. Cynthia, Associate Professor, to honor our dynamic principal, A. Alagappa Moses, Associate Professor and Head, Department of Environmental Sciences. I request Mrs. Shirley Elgal, Associate Professor, to honor our energetic Burza, Dr. G. Dhyanraj, Associate Professor and Head, Department of Commerce. I request Dr. Sharmila, Associate Professor, to honor our UG Head and co-convener of BICPAC'22. Thank you, ma'am.
Over to Ms. Leelavati, ma'am, Assistant Professor.
Our principal, Dr. Paul Dayabaran, a humble leader, has great passion for teaching. With immense vision, he leads Bishop Eber College, Trichy. We are happy and proud that he has been bestowed with AIACHE's Best Principal Award. Dr. Paul Dayabaran's firm attitude towards the academic rigor promoted the institution to rank 4 among arts and science colleges in India by the Ministry of Human Resource Development through NIRF National Institutional Ranking Framework. He is known for his wonderful administration and high achievements of the institution. His competence and ability enveloped with a mission lead the college to greater heights, like being recognized by the UGC as College of Excellence, the first among the affiliated colleges in Bharathidasan University to hold the title. Perplexed by his sagacity and administrative qualities, he is treasured by his subordinates, and as a teacher, he is loved by every student as he has molded and influenced the lives of several students by his kindness. Like the rainwater bringing life and lushness to the land, your words always ignites our souls, sir. May I now request our beloved Principal, Dr. Paul Diabaran, to deliver the presidential address and cast the light of his knowledge upon us.
Esteemed chief guest of this blended virtual as well as offline international conference, Honorable Vice Chancellor of Central University of Tamil Nadu, Thiruvarur, Professor Dr. M. Krishnan sir, beloved Vice Principal, Professor Adagappa Moses, beloved Bursar of the college, Dr. Nyandaraj, beloved Dr. Jasmine, the UG head of the Department of Biotechnology and Bioinformatics, and co-convener of this international conference, beloved organizing secretaries, Mr. Dinesh Raja and Dr. Vineet, beloved Dr. Ravi, the leading orthopedic surgeon of our Trichy town, learned deans of different disciplines, learned heads of different departments, my dear colleagues who are working in the Department of Biotechnology and Bioinformatics, my dear student friends who are pursuing UG Biotechnology, PG Biotechnology, and 5-year integrated bioinformatics, participants, students from other departments and other colleges, ladies and gentlemen, good morning. My warm greetings to all of you. It's a great joy and privilege for me to associate fraternity of biotechnology and bioinformatics as they thoughtfully organizing this 2-day international conference for the benefit of the student community of life science as well as other disciplines. First of all, I must appreciate and congratulate the UG head, the co-convener, Dr. Jasmine, and the active, vibrant organizing secretaries, Professor Dinesh and Dr. Vineet, and all the colleagues who are instrumental for this event to happen in our campus. Of course, we've been totally paralyzed by the pandemic COVID-19. It's named 19, towards the end of 19, in Western nations, it came into existence. It erupted throughout 20, 21, and 22. Till we have few cases. The past 4 semesters, students are in the campus for some time and off the campus for most of the time. And they had written all the end semester as well as internal tests, the past 4 semesters only through online. But you must take these as challenges. Whenever any trials and tribulations exist, we should not succumb to those. Always we must consider those as challenges or opportunities for us to think beyond and to excel ourselves. This morning we were talking in our room, our Vice Chancellor sir and Dr. Ravi sir, they have been talking. Dr. Ravi sir said, so far we did not find an apt vaccination for HIV, but this coronavirus infected the entire globe. The severity is much, much more when compared to other infectious diseases. Therefore, we also thought, we worked very hard, we were able to find the vaccination of different kind. That's the reason why we've been saved now. Dear friends, just like this, problems will always emerge. Problems will come in bundles, but we have to tackle one after the other. We should not get succumbed to those problems. We always consider those as challenges. In fact, your title is very appropriate, Biological Innovations and Computational Exploration for Pandemic Challenges. Very nicely phrased, and of course, plenty of course, I had that more than 300 papers have been submitted. Of course, apart from, it's very rightly planned. You have as many as 11 plenary lectures, 6 happening today and 4 will happen tomorrow. And out of 10 eminent scientists, 3 are from overseas, from University of Milan, Italy, from University of Edinburgh, UK, and from National University of Science from Seoul, South Korea. And we have 7 eminent scientists coming virtually to meet us to share their expertise from different parts of our nation. It's a great thing. It's a great opportunity for student friends. Just when we entered this dais, our Honorable Vice Chancellor said, during this pandemic time itself, you got a lot of students. I'm glad that all the students belong to maybe Department of Biotechnology, Bioinformatics. Of course, Professor Dr. Krishnan sir was Professor of Biotechnology of University. When he was here, our former Principal and Director, Professor Samira sir, introduced this program in early 2000.
the latest trends in biotechnology that we are all here. So I am very happy that a lot of students are pursuing this field. So as a doctor, I am seeing a lot of patients who have got sufferings which we are not able to completely cure. We are able to cure very few diseases. We are able to give only relief, symptomatic relief to some diseases and substitute some of the things which the body is not producing. But the aim is to cure every disease possible, and we should free the humanity from all these sufferings. So there is a great role, the role played by the doctors as well as the scientists. Only when both of us come together, it is possible to implement whatever is the latest trend and save the humanity from these sufferings. And pandemic has definitely proved that. So I really congratulate the organizers for making this event and inviting me to this. Best wishes to you. Thank you.
Thank you, sir, for your valuable words. We are about to listen to the reading of report of a conference. Organizing such a mega event like an international conference is an Herculean task, but God has been so kind and benevolent that he has been showering his blessings upon our department, and we are now able to organize in a smooth manner, much to be satisfaction for all those concerned. It gives me a delight to invite Ms. Leelavati, Assistant Professor, Biotechnology and Bioinformatics, to present the conference report.
Dignitaries on the dais, Honorable Professor Dr. M. Krishnan, the Vice Chancellor of Central University of Tamil Nadu, respected principal, eminent vice principals, revered bursar, respected delegates and enthusiastic participants from all over the world, dedicated members of the faculty and energetic students, a very pleasant morning to one and all. It is my immense pleasure to deliver the report of the DBT CSIR sponsored International Virtual Conference on Biological Innovations and Computational Exploration for Pandemic Challenges, BICPAC'22. The department organizes conference every year and has chronicled huge success in enlightening and enhancing the mental horizons by promoting the participation of brilliant minds. This year, our department organized an international virtual conference, and the purpose of this conference was to introduce and encourage young minds to explore new research ideas, induce thoughts, innovations, and find solutions to the new challenges in the field of biological sciences. We wanted to reach everyone, especially along the length and breadth of the nation. We, the organizing committee, started to send the brochure to all the academic institutions, research institutes, and NGOs from 19th November 2021. And the response was excellent. It was a pleasant surprise and blessing that our department had the privilege of receiving the financial assistance from the prestigious Department of Biotechnology, Government of India, and Council of Scientific Industrial Research, Government of India, for conducting the 2 days international virtual conference. We are happy to state that the response obtained was overwhelming, and we have received 316 abstracts from all over the nation, among which 180 were selected for oral presentation
Nanaraj, Dr. Ravi, and most of my students are working here. Maybe Anita took more advantages to read my biodata. Thank you very much. And also that management of this great college. Whenever I am visiting this college, there is a life, I can say. Even I can say that today, even the Central University of Tamil Nadu, it's difficult to bring the students inside the campus. Why? Because of the COVID. But I could see that 1,000 students are here to see that one side book fair, another side science exhibition, another side a conference on biotechnology. That is the greatness of the principal who is very active all the time. Though he's having his own problem in his health, but never see that. I see one such a principal who came to my small house and invited me to be here for this conference with the organizing secretary. So I bow my head in front of you, sir, for giving the respect on us. My dear students, this is a great occasion for you to attend such a wonderful conference. That is the international conference on biological innovations and computational exploration for pandemic challenges. Actually, before I am going to give my inaugural address, I want to congratulate the whole team, but I take two representatives to give or the honor the particular persons, secretaries of these two conferences. Maybe I can go with the Ponnadai for the two organizing secretary, Mr. Dinesh, and also that living. My dear students, I think you have the Dr. Ravi, who is a very, very busy doctor in this Tiruchirappalli. Why he is here? He always looking for innovation in science. Actually, he is having the lab on stem cell technology opposite to your college. Already you have the MOU with this laboratory. But before I am coming to this topic, I want to discuss some of the things. I request that my dear young students, don't take it as a conference is a festival or celebrations. Kindly take this as a challenge. What today or the tomorrow you are going to learn from the experts. Because this is a very good opportunity for you to see that lot of experts from various parts of that globe. Even I can see some of the persons is going to talk from Edinburgh. So what you have to do that, it's not possible within 2 days. Deliver everything, it's not possible. But kindly get into the subject and then explore it. What the achievements we are having in this particular field. Even Dr. Ravi and my dear principal rightly discussed that even though we have the HIV, we are crying on HIVs, but still now we don't have the proper vaccine for that. But within 2 years, we have the comfortable vaccines for that coronavirus. So that is the achievement. Even I can say that when the 1990, when the whole genome sequence of the human is going on, that time there is a lot of criticism from that throughout the world. Why they are spending this much of money? Even some of the scientists from India, they are participating in this program. They don't want to give the money, or they don't want to get that. I mean, it's not possible to get the grant from the ministry, because what it's going to do that. But today, the human genome is playing the major role. Even today, when I am asking my students, most of the students, they don't know that how many genes you are having, how many proteins you are expressing in your body, how it's going to play on the role. No. So what I want to do, that this is the right time. Kindly get into the subject apart from your curriculum. Yes, I accept it. We have to learn the curriculum. We have to get the mark and get the placement. But apart from that, a common knowledge is very important. The day I can recall that, because I can salute Biotechnology College. Why? Because this is the one among the college to provide the opportunity for me to deliver my science in this department. Even I can recall that our great leader, Swamiraj, was there when I am coming here to approve the program on bioinformatics. But actually, sorry to say that time I denied. I don't want to give the bioinformatics to the Biotechnology College. Why? Because they don't know the computers, they don't know the facilities. So I told that it's not possible to get the bioinformatics in this college. That time principal was completely worried, and then took me to the Swamiraj room, I think nearby that the next building. He told that, sir, he's my friend, but he doesn't want to give the bioinformatics to the Biotechnology College. I can recall the day, recall the word from Swamiraj. I know Dr. Krishnan. He's a very strict person in the university, transparent. So whatever he says, kindly follow that. I am not recommending bioinformatics this year. This is the word Swamiraj told that. What I took it in the other way, such a leader you want that, sir. So next year he developed the facility. He called me again for the commission. I had given the bioinformatics along with the Parthasarathy. So that is the way a college should develop. When after that I am frequently coming to this college, but I don't know that time Madam Violet was the head of the department of this department. I don't know that he's the wife of the principal. I scolded in so many ways, but he politely listened the things and did the marvelous things in the department. So what I want to say that is the way a department should grow. That is the way you have to bring the facilities. That's the way you have to bring that things to the students. So I have to congratulate the principal, earlier principal, administrator, everybody. Now, I frankly say that we are closing the bioinformatics in Madurai Kamaraj University, even though it's recognized as a school of excellence. We didn't get the students, but here they are running 5-year integrated bioinformatics program. I don't know that what's the secret behind it. I don't know that. Because last week I have the meeting with our chancellor. I think you know that great personality, Dr. Badmanaban. When we are discussing about it, because I want to start a new program in MSc Bioinformatics in Central University of Tamil Nadu. But my chancellor rightly, no, don't start the bioinformatics. We are not getting the student. So kindly start that proteomics and genomics. So I have to learn so many things from this Biotechnology College, how I can get more students for the bioinformatics. Anyway, before I am going to talk about the topic, the biological innovation, computational exploration for pandemic challenges, my dear students, Now everybody is talking how we are very fast to develop the vaccines. Kindly, 4 things, kindly keep it in your mind. Today, that is for the achievement. Number 1, you should have that cooperation with the various institutes. Please listen very carefully. You should have collaboration and cooperation with the different institutes, with the different experts of different subjects. We need basic science, advanced science, statistics, medical doctors, computational scientists, economics, and everybody should come together to start that or to go for such wonderful studies. That is the number 1, you need collaboration. Number 2, very importantly, you have to come out with a boundary. That is a very, very important thing. What I want try to tell that now this type of conference is going to tell that how I can come out from the Bisabibar College. Because whatever the research you are doing in Bisabibar College, it's not going to give much. Now I know that you are bringing the experts from different parts of the country and the globe, which is going to tell that how I am going to come out from the boundary of Bisabibar College and collaborate with the different people in different part of the country. That is the second point. Third point, sustainability. Starting any program without that, what you call sustainability, it's not possible to win the race. You have to have the sustainability. Now, you see that coronavirus, particularly we are talking, we reach the sustainability at least. Facilities, students, expertise, everyone should come together. Then only you can do that. Then 4th one is globalization. Everybody is thinking that the globalization is to bring that foreign students inside the country or our students are going outside the country. That is called globalization. No, it's not. How we are going to compete with that international bodies. That is a very, very important thing. Kindly recall that today, Bharat Biotech produced the vaccine for the COVID or the 19 to give the vaccine to the other countries. My dear students, maybe it's not out of that our conference. What is the higher education? Higher education should reach the society. Kindly make it very careful. Our leader Mahatma Gandhi rightly told that when you are not taking my higher education product to my society, then I don't want to respect the higher education. Why I am telling that, though we are having 1,000 universities in India, 40,000 colleges in India, 200 research institutions in India, but none of that institutes, college, or the university are not reaching the global ranking. Why? Why? We don't know the scientists in India. We don't know the money in India. We don't know the experts in India. We have everything. But we forget to take the excellence of higher education. Excellence of higher education. Innovation in higher education to my society. That is a very, very important thing. My dear young students, please listen very carefully. Maybe you can get that biotechnology degree here. You can get a higher post here. You can draw good salary here. You can live very happily. No, that is not the life at all. What you are going to give to my society, to the village, to the parents, to my relatives, that is that higher education. Don't forget about it. So only starting from our Honorable Prime Minister Modi to the Honorable Chief Minister of Tamil Nadu is telling, don't forget the villages. Don't forget our culture. Don't forget our tradition. So that is the higher education. I am sorry to say, whether we are following these 4 important points, think about it. Recall that, how we are going to enter into that. Now I am coming to that, what you called your topic. What do you mean? Biological innovations and computational exploration. Particularly when you are talking this particular heading, without a computer science experts, we are not succeeding this attempt. Because what I am going to tell 10 topics, but if you want to talk about each topic, we need a separate session. But I briefly tell that. Kindly get into that. Google or whatever the website you are having and get more ideas how you are going to have it. Number 1, artificial intelligence. What you are going to do with artificial intelligence? My dear students, whenever you are going to get something, because the day I can recall that 2019, Wuhan in China, where we got the information. There is a coronavirus. The whole world, it's going for the lockdown. Even Madurai Kamaraj University, when I was there, exactly March 24th or April 1, we closed that. So just started. But how I can bring the whole data, where that coronavirus infected, how it's spread, how the people are affected, and how I can collect the data. That is that artificial intelligence. But every college, every university we are talking. The 2 months back, we have that the topic on artificial intelligence. But there is no experts. I think my biotechnologist, my bioinformatics, kindly look into that. Where is that experts in artificial intelligence? So artificial intelligence is going to give the lot of data without reaching the people. One example say that even the people are talking too much about the artificial intelligence. But Thiruvananthapuram, one mall, completely working on artificial intelligence. I don't know how. I think you are enter into the mall and then you can take anything from the mall, whatever the items you want. And then come to the counter, you give the card, they will collect the money, you can go away. Nobody is there. Nobody is there. That is a completely computerized and what you called having that, chips everywhere. And then they will collect the money. The same thing. Now artificial intelligence help us to collect the data throughout the world. Throughout the world. Now everybody is talking how many people affected, how many people are hospitalized, how many people are discharged from that coronavirus. Everybody is talking about it. That is what you call artificial intelligence is doing. But without the computer knowledge, you can't operate it. Second one is blockchain. I don't want to enter into your politics, but blockchain is very important. What it's going to do that? The blockchain is going to do that. Provides the transparent and the cheapest way of transfer the medicines. There is no what you call in-between brokers, direct contact. I think you also can recall that. First, when we people are going for the vaccination, they charge 850 rupees. Because of that, I don't want to get into that detail. But why? Why? But later now, everywhere free of vaccines. Why it happens? But other countries' blockchain played a major role to give that production of the vaccines very easily and then get into the people very easily without any in-between brokers. But the only one disadvantage is there. When you are going to have that money exchange, where is the drawback is where is server will be available, how you are going to keep the secret of that handling. These are the things are there, but any war, you are having some difficulties. But how we are going to come out is very important thing. That is the science. That is the technology. For that blockchain, you need the computer science expert. Number 3, open access technology. Otherwise, you can talk about open source technology. Imagine that when I am doing a research in Bharatdasan University, in the Department of Environmental Biotechnology, when I am not publishing my result, how I am going to get that the discussion? My dear students, you know that within the short time, 183 sequence of variants of coronavirus available. That is published in your Springer or whatever you want to call that. It's available. So if an Indian wants to work on that, the sequence, immediately he can access the data. There is no secret on that. That is called open source of technology. My dear young students, boys and girls, kindly keep the topic in your mind. After the conference, when you are going to your respective area, collect the information. How much it's available, how much data available, how it's useful to the society. Number 3, now first I told that artificial intelligence. Second, I told that blockchain. Third, I told that open source technology. If everything is available with you, now I am going for telemedicines or telehealth. I think doctor will also accept it. Even yesterday TV, I am seeing that instead of going to the hospital, the medicine is coming to your house. I can recall that the earlier Chief Minister of Andhra Pradesh, Chandrababu Naidu, why he got that very good name in the agriculture sector, he had given that what you called your iPad to all the agriculture officers along with the farmers. So when the farmers is going to identify any disease in my plant or in my agriculture area, take the photograph, give to agriculture officer, immediately they will give the solution. No need to wait. The officers should come, they have to collect the data, then identify the solution for that. No. That is now we are doing it. As my principal rightly told that when the pressure comes, we will work. Yes. Because thousands of people are dying. What I can do that? I have to work together. I have to go for that each and every house to give the medicine. Then only we can cure the people. That is called telehealth technology. Imagine that. A biologist can do it themselves. It's not possible. Or a social scientist can do that themselves. No, it's not possible. Mathematician can give some algorithm without a computer science people. No, it's not possible. So I told that now the title is going to speak. Bioinnovations, as I told that, excellence and innovations. That it's going to help you to go for that public directly. That is, we need the computer science exploration. Then I can say that now medicine comes. One side, everybody wearing the mask. Somebody is identifying the different instruments. How you can check it? How you can explore that, whether it's worked out properly or not? That is called 3D technology. Otherwise, they are simply called 3D printing. So layer by you can layer, analyze it. How my vaccine is working, how my mask is working, how the diagnostic hit is working. So the 3D technology is going to tell about it. That is the next technology. You should learn about it. Then sixth one is very powerful. Very powerful. Gene editing technology. Gene editing technology. My dear students, please give your attention. As I told that, it's not a celebration. It's a scientific discussion. So what is there? Kindly get into the science of coronavirus. How it's entered into the body. How it's infected to the different parts of the body. Every organ is having the receptors. Because of the spike protein available in your coronavirus, it's going to do that. It's attached to the receptor. CSIR3. So how it's going to attach and how it's going to spoil our organs. But what the gene editing is going to do that, kindly break the attachment. Any bioinformatics students are there? Kindly look into that, study that. What is that, what you called that, interaction, protein or that ligand and receptor interactions. You can break it. You can break it by gene editing. I don't know, it's not a new subject, but very expansive subject. 1957, where they discovered the technology very, very deeply manner. That is called, I think, I don't want to tell that, but my teachers can explain that, CRISPR technology. Where your gene editing can do that. That is that next one, what I can say that gene editing technology. So if you are giving time, maybe 2 hours we can talk about the gene editing technology. How your gene is going to do all these things. The receptor interaction with the protein. So in Bharatdasan University, we also did it. Some of the work receptor, interaction with the proteins. Then next one is today, synthetic biology. Synthetic biology. What do you mean synthetic biology? Why you need that? Without touching that my microbes or virus. By having the gene sequence. Because as I told that, more than 100 coronavirus sequence already done it in different countries. How I can get the live samples? It's very difficult. So what I can do that, I can synthetically prepare the sequence. I can activate in my lab, and then I will work on that, how it can be prevented. How it's, spread throughout India. So these are the things we have to do that. Then I think when I am going for the NAC accreditation, every university is doing that. I am developing the robotics. Everybody is saying that robotics. But what happened? Whether the robotics came to Bishop College, or it came to Central University of Tamil Nadu, or it came to Tamil Nadu. No, where your robotics is working. But now, every corner of the country, because everybody is afraid of that. Whether you believe it or not, the April 14th, I can recall the day, in Madurai Kamaraj University, we can got some, I think, unexpected data. I don't know how many of you know that Madurai Kamaraj University. It's surrounded by 75 villages. If one person affected by corona, the whole village is afraid. They are psychologically affected. How I can give the medicine? How I can save the people? We need robotics. We need robotics. The robotics can move with the medicines and solve the problem. I think now we got the collaboration, because you know that not for coronavirus, I am telling. We know that how many of you people visited Thiruvarur. There is a very big tank is available near Thiruvarur temple. So far, it's very difficult to clean. So we are having that, or we are collaborating with the German technology. They are not going to do that. The robotics will come and clean the tank. So that's the way. Now the robotics reaching that each and every corner of that house and give the medicines. But you can ask the question, sir, what happened to the robotics? It's a machine. We are accepting. But is it not infected and spread that? But this is completely what you call de-infected by that ultraviolet. So that's the way now robotics is working. Finally, I can come that one technology called drones. If you are asking my young students, had you seen the drones? Yes. In my marriage function, they are coming taking the photograph. But today, the drones are going to be used to spray the medicines, disinfectants, take the medicine one place to another place. Right? So, my dear students, I told you 10 technologies. Analytics, that's artificial intelligence. We need instrument to work. Blockchain, open source technology, telehealth technology, 3D printing, gene editing, synthetic biology, robotics and drones. 10 topics I discussed, or I give the brief idea to you. But in the nutshell, imagine that without a collaboration with the computer science people, without the collaboration of engineering people, without the collaboration of my basic science people, can I succeed in this attempt? That is called biological innovations in and also computational exploration. That's only I told that. The first itself I told that. Kindly have the collaboration. Kindly come out from the border. Have the sustainability and also globalization. Sir, finally I want to tell, take that the big companies of computer applications or your internet area, starting from Sundar Pichai to Krishna, IBM. They are leading as a CEO in foreign countries. Kindly tell that. Kindly put your hand on your heart and tell that. What is your innovative product available in India? Because every university we are saying, we are innovatively working. We are having the incubators. Everything we are having. But where is the product? My public is longing for you. Solve my unemployment problem. Solve my electricity problem. Solve my drinking water problem. Are you giving the solution for that? That is the thing, my dear young students, we are expecting. Please, when you are going to work, of course, now I have near about 200 students here. How I am going to differentiate my students from one among other? Everybody is having MSc Biotechnology, MSc Bioinformatics. But I am looking for the skill. I am looking for the skill. Learn that apart from your curriculum. Learn your skill, develop your skill. Then only succeed in your life. Because today, the world is more competitive. Rather than competitive, it's having the challenges. So if you want to live in this competitive world, if you want to take the challenges, apart from your curriculum, you need innovations. You need skill. That is the thing I want to tell that finally, I think we are all feel that we are the superior of the world. We are the superior of the world. Human is the superior of the world. But today, a small virus is teaching to us how to live in this country. How to take care of the environment. How to take care of my life. So everybody is equal. That's the way. Now we have to learn. I have to thank the coronavirus. We are learning so many things. Now only we are respecting our elders. Now only we want to live with my elders. Now only we want to live with a joint family. Because we are affecting. Whether corona is coming to me tomorrow, day after tomorrow, or what I am going to do that. So that's a life we are going to do that. But I am so happy because of that. When I was at Madurai, when I entered in the 19th January, I can see 10 or 20 peacocks. But after the coronavirus, I have 200 to 300 peacocks in my campus. Why? Because human movements is not there. We are not disturbing the environment. So why not? We can keep on going to take care of the environment and bring this a beautiful earth to my future. By that, youngsters, you are the brainy people today. You have to take our country with the high flag in the world scenario. With this, have your life. Maybe tomorrow you are a good scientist, good leader, good entrepreneur, whatever you are. But be a human being. Be a human being. And also following the ethics in your life. With this, I wish you all the best for the great conference. I think you can learn a lot from the people coming from the different part of the country, world. And also, you have the fruitful time in another 2 days. Thank you very much for giving the opportunity.
If your action inspires others to dream more, learn more, and become more, You are a leader, says John Quincy Adams. At this juncture, I would like to attribute this proverb to our chief guest. Thank you so much, sir, for your informative and thought-provoking speech.
We would like to record our deep sense of gratitude to the esteemed judges of the scientific sessions. Now it's our time to honor all the judges, the dedicated personalities to be heartily thanked for. Over to Dr. T. Weston, Assistant Professor, Department of Biotechnology and Bioinformatics.
Now, I request our Honorable Vice Chancellor to honor the judges of oral and poster sessions with a memento. Please, sir. Dr. Anand Gideon, Associate Professor, Head of the Department of Botany, Bishop Keeper College, Trichy. Dr. Priscilla Suresh, Assistant Professor, Department of Zoology, Bishop Keeper College, Tiruchirappalli. Mr. Jeremiah, Assistant Professor, Department of Zoology, Bishop Keeper College, Trichy. Dr. Nesarajan, Assistant Professor, Department of Zoology, Bishop Keeper College, Trichy. Dr. Paul Ajit Kumar, Assistant Professor, Department of Botany, Bishop Keeper College, Trichy. Dr. Emmanuel Sagairaj, Assistant Professor, Department of Botany, Bishop Keeper College, Trichy. Thank you, sir.
Thank you, sir. We are now at the end of this inaugural function. Before we close, we would like to express our sincere appreciation to all of you who generously helped us for organizing such a huge event, thus making this conference come together. I now request Mr. J. Dinesh Raja, Assistant Professor, Department of Biotechnology and Bioinformatics, and the Organizing Secretary, BICPAC'22, to render the vote of thanks.
Praise the Lord, O my soul. Praise his holy name. Praise the Lord, O my soul, and forget not all these good things. Honorable Vice Chancellor, sir, respected principal, respected bursar, respected members of the faculty, respected delegates, and my dear students, judges from the different departments. I thank the Almighty for giving me this greatest opportunity to be as one of the organizing secretary in this international event. On behalf of the Department of Biotechnology and Bioinformatics, it's my duty to thank our Honorable Vice Chancellor, Dr. Krishnan, sir. You know that Central University of Tiruvarur is one of the notable institutions among 54 central universities. It comprises of 12 schools and 27 departments. As we know that, being an administrator, being a Vice Chancellor of the Central University, how the Vice Chancellor is busy must be. And though his busy schedule, I humbly thank our Vice Chancellor, sir, for giving the consent to be the chief guest and deliver the inaugural address. Thank you very much, Vice Chancellor, sir. I thank our principal, Dr. D. Paul Dayabaran, sir, and also our Vice Principal, Dr. Alagappa Moses, sir. Whenever we approach regarding the conference work, like getting consent with speakers, application process to the DBT, CSIR, and central agencies, applying for the ISB number, clearance approval from the Ministry of External Affairs. So our principal and vice principal helped us with great support and motivated us all the way. With all your support, it is not possible to conduct
this event in an efficient manner. Thank you, Principal Sir. I thank our respected Bursar Sir. He's the person who is responsible for all the financial matters in the institution, helped us and supported us in all the way. Thank you, Bursar Sir. And I thank our coordinator, Madam, Dr. V. Violet Dayabran, for her support and prayers, always for the betterment of the department. I think ma'am is watching us in online. So thank you very much, ma'am, for all your prayer and support. I thank our UG coordinator, Dr. R. Jasmine, ma'am. From the day one, starting the conference, ma'am is rendering her full support and freedom to us in all the way. Thank you very much, dear elder sister, for all your prayers and support. I thank the members of the editorial team. It is not an easy task to receive 316 abstracts from all over the nation, compiling, separating as oral and posters, allotting separated room for the delegates to do their oral and poster presentations. I thank Dr. William Raja. William Raja, are you here? Please. Dr. William Raja. And I thank Dr. Nyanraj. Nyanraj sir is here. I thank Dr. Nyanraj. And I thank Dr. Bliss, Ramya ma'am, and Dr. Miss Leela ma'am, assistant professors, for their hard work and effort to bring out this event well. Thank you, professors. I must thank my brother and also assistant professor, Dr. Benjamin Tamilselvan, assistant professor of zoology, for helping us in applying DBT, drafting the application through this conferences. He's working as a neighboring department. I thank all my brothers and sisters in the biotechnology and bioinformatics family. Without your support, it is not possible to conduct this grand event. I thank our dear student friends. You made this event successful. The delegates. The success of the scientific conference is not based on the number of abstracts received. I thank you, dear delegates, for your active participation and all part of the nation through online. I thank the sponsors, DBT and CSIR, for their financial support to do this event with your recognition and support. Once again, on behalf of the Department of Biotechnology and Bioinformatics, the organizing committee, thank you very much for all your support and prayers. Thank you very much.
Thank you, sir. Our hearts are inspired with joy on your readiness to be with us. Today, dear guests present on and off the stage, you are so precious to us. We would like to save this moment. We would now hold a photo session. We kindly request the dignitaries on the dais to take a position.
I kindly request the faculty members of the Department of Biotechnology and Bioinformatics to come over to the dais for the photo session. Thank you.
Once again, to one and all gathered here on this virtual international conference. Let's start with the next session on this first day of virtual conference. Starts with the first plenary lecture. Starts with the speaker.
I invite Dr. A. Anita Margaret, Assistant Professor, to give a brief introductory about the respected speaker, Dr. Sanjay Deshmukh, former Vice Chancellor and Professor of Life Sciences, University of Mumbai.
A pleasant good morning, good afternoon indeed. Dr. Sanjay Deshmukh, Professor of Life Sciences and University of Mumbai, is an illustrious academician and researcher fortified with more than 2 decades of professional proficiency. His area of research expertise is agricultural biodiversity, genetic resources conservation, and the sustainable management. Contemporarily, he pioneers on mangrove ecosystem that led to establishing global network of mangrove genetic resources conservation centers. He has developed people-centered hotspots strategy for conservation planning through extensive first-hand study on sacred groves in Maharashtra. Holding a profound working experience, Sir instigated his career as Professor of Life Sciences at the University of Mumbai since 2005 and has served as the youngest ever Vice Chancellor at the University of Mumbai, which has a heritage of 162 years. To highlight his academic credential, he is the only research advisor recognized with PhD guideship in 5 subjects: geography, botany, life sciences, environmental sciences, as well as biotechnology. For his credit, Professor Sanjay has 172 research publications in recognized journals, which augments his excellency in research. He has implemented 32 research projects with total research grants of over 17 crores. His achievements are boundless and is not restricted within India. Yes, being a passionate researcher, he has traveled around 45 countries and has been recognized by several pioneered institutes globally. All his accomplishments are recognized and certified by a cascade of awards. To name a few, Talented Scientist Award by International Society of Research in Medicinal Plants and Herbal Products, Johns Hopkins University, Maryland, USA. The Maimar-e-Watan, a civilian award of the Sindh province of Pakistan. Best Village Development, an NGO category award constituted by the Ministry of Environment, Forest and Climate Change, Government of Maharashtra. Sir has been a research fellow in various national institutes such as NISA, INSA, and LEAD. Such a multifaceted and eco-conservational person who focuses more on environmental sustainability is here with us. We are thankful, sir, for accepting our invitation and find you as an optimistic choice to serve as a resource person in today's session. I welcome you, sir, on the behalf of our department. Thank you for being with us. Thank you.
Good afternoon, everybody. Am I audible? Hello?
Yes, sir.
Am I audible?
Yes, sir. Please, you can proceed your plenary lecture, sir.
Yeah. Thank you very much. And I'm really grateful to all of you for providing me with this opportunity. I have been tracking the morning session and I was privileged to listen to Honorable Vice Chancellor, Professor Dr. M. Krishnan, the Honorable Vice Chancellor from Central University of Tamil Nadu. I really congratulate him for his visionary address, which has set the tone for this important event, which is viewed not only from within India, but also from outside India, from several countries. So respected Honorable Vice Chancellor, respected Principal Dr. Paul Dayabaran, who is the Principal of Bishop Heber College, Tiruchirappalli. We have distinguished speakers, and in the first session, we will be having Dr. Kumar Somasundaram from IISc Bangalore, along with Dr. J. Jayakanthan, who is from Alagappa University. There are eminent speakers and participants, both scientists, teachers, and researchers, as well as students, who will be making a presentation of their research in this important event. Besides, there are very many observers from different parts of the world who are watching this important event. I am particularly delighted and extremely privileged to be a participant by way of sharing some of my thoughts in this important event organized by a unique college in Tamil Nadu, an autonomous institution, that is Heber College, and the Department of Biotechnology and Bioinformatics, which is organizing this event. I congratulate them. This international virtual as well as hybrid conference on biological innovations and computational exploration for pandemic challenges is a two-day event, and it's very important for us to look at the current situation brought by pandemic and the future, at least during the 2 years that we have experienced, is going to be volatile, uncertain, complex, as well as ambiguous. And therefore, it is very important for us to look at science and technology that should always benefit society from the perspective of revolution in biological sciences, in technology, and in many areas, including social sciences, as well as economics and bioinformatics. These innovations from the point of view of technology, the renewal of technology, and the innovations, the newer ideas culminating into the technologies, both of them are very important. And therefore, I would like to put forth some thought on an issue of biorevolution, and that can be achieved through both biological transformation and technological innovation. This is the theme on which I'm going to share some of my thoughts with all of you. In fact, if you look at the post-independence era of our country, then almost 7 decades, which coincide also when the double helix structure of a DNA molecule was discovered. And after so many years of such discovery, the world of biology appears to have reached a new phase of growth. A flurry of recent innovations, for example, CRISPR-Cas9 to edit genes and stem cell advances to reprogram cells, they're all providing new understanding, new materials, and new tools, in addition to the cost at which they're developed are much less than what was envisaged before. And therefore, when we look at the advancement of science, it is so much advanced. For example, a few years ago, particularly in 2016, a human cell atlas project was kicked off to create comprehensive reference maps of all human cells as a basis of research, diagnosis, monitoring, and treatment. As a result of that, this particular scientific advancement, a growing number of applications are emerging from the lab, and they are all being put to commercial use. The potential of beneficial economic and social impact seems enormous. As much as 60% of the physical inputs to the global economy could, in principle, be produced biologically. Our analysis suggests that around 1/3 of these inputs are biological materials. For example, wood, cotton, and animal bread for food. For these materials, innovations can improve upon existing production processes. Again, to give an example, squalene, a moisturizer used in skincare products, is traditionally derived from shark liver oil and can now be produced more sustainably through fermentation of genetically engineered yeast. The remaining 2/3 of the materials are not biological technically, and the examples of these can be mentioned as plastics and aviation fuels. But of course, they could in principle be produced using innovative biological technologies or processes, or can be also replaced with sustainable practices like substitutes like bioinnovatives. When we are thinking about all these things, an example of that comes to my mind is nylon, which has already been made using genetically engineered microorganisms instead of petrochemicals. To make things little clear, reaching the full potential to produce these inputs biologically is a long way off, but even modest progresses toward this particular phenomena could really transform supply and demand and economics and participants in provision of physical inputs. Biology has a potential in the future to determine what we eat, what we wear, the products we put on our skin, and the way we also build our physical world. Utmost importantly, in the 21st century, where the advances are aimed at, is human health. And human health is one of the most significant domains where biological advances are being applied. The biology is already being saving lives through innovative treatments tailored to our genomes and microbiomes. I'm sure in the future, when we're talking about technology development, we are estimating that almost half of the global disease burden could be addressed through applications that are scientifically conceivable today. Moreover, many of the innovations born of these bioinnovations contributed to the global response of the SARS-CoV-2 pandemic in early 2020. There are many other domains, from agriculture to energy. They could also benefit from biological processes and products. Biology could even be deployed to mitigate climate change and helping reduce the man-made greenhouse gas emissions. However, the risks from these innovations are profound and unique. Biological systems self-replicate and are also self-sustaining and are highly interconnected. Changes to one part of a system can have cascading effects and unintended consequences across an entire ecosystem or species. Accidents can have major consequences, of course, and particularly if used unethically or maliciously. Manipulating biology could become a Pandora's box that, once opened, unleashes lasting damage to the health of humans, ecosystems, or either of the two or both. These risks are particularly acute because of many of the materials and tools are already cheap and accessible. Moreover, tackling these risks is complicated by a multiplicity of judicial and cultural value system, which makes collaboration and coordination across countries extremely difficult. Bioinnovation, bioinnovation is occurring in different key areas, and I can name at least 4 of them. A wave of innovation is being enabled by advances in biological sciences, which are accelerated by developments in computing, data analytics, machine learning, artificial intelligence, and biological engineering. If you want to group the innovations into 4 arenas, then they could be grouped into biomolecules, biosystems, biomachine interfaces, and biocomputing. Major breakthroughs when we are talking about biological innovation, then in each of these areas that I have mentioned, that is biomolecules, biosystems, biomachine interfaces, and biocomputing, in each of these 4 arenas are reinforcing one another. In biomolecules and biosystems, advances in omics and molecular technologies, that is the mapping and measuring of molecules and pathways within cells and engineering them, are enhancing our understanding of biological processes as well as enabling us to engineer biology. I can give an example. CRISPR, that is CRISPR technology, allows scientists to edit genes more quickly and precisely than previous techniques. Advances in biomechanics and biocomputing both involve deep interaction between biology and machines, which is becoming increasingly possible to measure neural signals and power precisely neuroprosthetics. It is now also possible to store the world's wealth of data using DNA. By some measures, 1 kilogram of DNA could hypothetically store all current data in the world. In addition to that, new biological capabilities could also bring about transformational change in economies, societies, and also our lives. To be precise, the new biological capabilities have the potential to bring sweeping change to economies and societies. The effects will be felt across value chains, from how R&D is conducted and how the experiments are conducted, to the physical inputs in manufacturing, to the way medicines and consumer products are delivered and consumed. The capabilities can be highlighted in few points, and I would like to mention these points, such as biological means. The biological means can be used to produce a large share of global economy's physical materials, potentially with improved performance and sustainability. The increased control and precision in methodology is occurring across the value chain, from delivery to development and consumption, with more personalization. Increased control, for example, in methodology and precision in methodology, is occurring across the value chain, from delivery to development, and the capability to engineer and reprogram human and non-human organisms is also increasing. In addition to that, new methodologies using automation, machine learning, and proliferating biological data are enhancing discovery, throughput, and productivity in research and development. Potential in these technologies is also growing for interfaces between biological systems and computers. A new generation of biomachine interfaces relies on close interaction between humans and computers. While these are early days and a prediction of a specific development or enhanced innovation may be little difficult, but the scope and scale of these emerging capabilities could have a broad impact on economies and societies, in addition to touching multiple domains, both directly and indirectly. These applications may change everything from the food we consume to textiles to the types of health treatments we receive and how we build our physical world. The potential value of these innovations is vast. And as I mentioned a bit earlier, as much as 60% of the physical inputs to the global economy could be produced biology, and even modest progress towards that 60% number could be transformative. Beyond the physical world, innovations could transform prevention, diagnostics, and treatment of diseases. Here, I would like to mention that at least 45% of the global disease burden could be addressed with capabilities that are scientifically conceivable today. A visible pipeline of applications can also deliver profound impact across a wide range of domains within the next 2 decades it is envisaged. When we talk about human health and performance, they have the most scientific advances and the clearest pipeline from research to application. The science is advanced, and also the market is generally accepting the innovations which are coming our way. However, based on our use cases and the experiments that have been conducted by the scientists and technocrats and policymakers, more than half of the direct impact is likely to be outside health, primarily in agriculture and consumer products. More than half of the impact from the visible pipeline of applications is also outside healthcare, particularly in agriculture and other areas. I would like to mention about 4 domains which I had briefly highlighted a little while ago. The most of these value in the next 1 or 2 decades will come in these 4 domains or the cluster of sectors where applications are emerging from bioinnovation. We would like to mention here a summary of these 4 points. And the first point obviously is going to be a very important domain, that is healthcare and performances. A new wave of innovation is underway that includes cell, gene, RNA, and microbiome therapies to treat or prevent disease. Innovations in reproductive medicine, such as carrier screening or improvements to drug development and delivery. You will be astonished to know that the direct annual global potential impact in this particular domain, that is health sector, is estimated around US dollars 500 billion to about US dollars 1.3 trillion over the next 10 to 20 years. And it can be also mentioned, or parallel, it can be predicted that 35% of the overall impact that we are estimating for this particular period out of any innovation. When we talk about agriculture, that is the second domain, agriculture, aquaculture, and food, where applications such as low-cost, high-throughput microarrays have vastly increased the amount of plant and animal sequencing data, enabling lower cost or low-cost artificial selection of desirable traits based on genetic markers in both plants and animals. This is also known as marker-assisted breeding and is many times quicker than traditional selective breeding methods. The third domain is obviously the consumer products and services, where opportunities are opening up to use increasing volumes of biological data to offer consumers personalized products and services based on the biological makeup. The applications include direct to consumer, that is DTC, genetic testing, beauty and personal care based on microbiomes, and innovative approaches to wellness and fitness in both humans and pets. When it comes to materials, chemicals, and energy, the new biological ways of making and processing materials, chemicals, and energy could transform many industries and our daily lives, although the economies are challenging. Improved fermentation processes, for example, can increase the speed of production or quality of materials that are already created using fermentation, such as food and feed ingredients. In this way, biology has many other potential applications, although some of these are likely to be further in future. It could be deployed to help the environment through biosequestration using biological processes to capture carbon emissions from the atmosphere, and bioremediation, which is a process to remove inorganic and organic compounds from soil, water, and atmosphere that might be harmful. Other potential applications of biological innovations, they could be found in education, defense, and even space exploration. While we expect that biomolecules and biosystems innovations will drive the largest direct impact across the range of domains, the impact are also emerging in bio-machine interfaces and biocomputing, where the science and development are at an early stage by applications, but these applications are very much promising. The applications that have already been developed, including neuroprosthetics to restore hearing and vision, are noteworthy. This is the first theme of the international conference. And when we talk about the second theme, that is the biorevolution through technological innovations, I would like to put forth my thoughts in next few minutes. As all of us know, and it has been well understood, the dynamics of COVID-19, but it has also caused immense impact on hospital systems, businesses, schools, as well as economy, telemedicine, telework, and also online education. It has become very much essential because these are the technologies which we have been talking about, but which we never used, like telemedicine or teleworking and online education. So they have become very essential to help societies to slow down the spread of the coronavirus. The pandemic has also generated a very rapid impact with respect to the technological advances that we are talking about, and also a demand that has been generated for efforts to use these innovative technologies to cope up with damage from COVID-19 on our life. The pandemic has not only raised opportunities to advance technology-based solutions, but also have provided a rare opportunity to all of us to study the research and practice of technology, including information management, work practices, and design and use of technologies. The quick transition to telehealth, telework, and online education in response to the coronavirus threat is a reminder that digital technology brings many benefits and play an essential role in managing and reducing the risks caused by the lockdown during pandemic and even after the pandemic. Just a couple of days ago, we had Honorable Prime Minister of India talking about digital meta university, a concept that we have been discussing for a decade almost, but nothing had happened because a lot of preparation had to go. And now we are talking about making digital university a reality by middle of 2023. And the Honorable Minister, as well as the chairman of AICTE, the chairman of UGC, they gave their views and committed themselves to help the country in getting into this phase of establishment of digital university. So these are the advances which we have been forced to look for, think for, and as solutions to the challenges posed by pandemic. And therefore, it is very important to understand, which is of course well known, that information systems and information technology, they can play a very important role in healthcare clinical decision support, emergency or crisis response, and risk management. This is just one domain that I'm talking about, application of IT innovations in healthcare. The existing IT solutions which are prevalent are very much there, like mobile COVID-19 contact tracing applications, which are now, of course, developed for the last 2 years, and chatbots, they have been recently developed to fight the pandemic. Application of these technologies can help reduce the impact of the coronavirus pandemic on people, organizations, as well as society at large. Effective and innovation that we are talking about, the innovative use of emerging technologies can help identify community spread of the coronavirus, monitor the condition of the infected patients, improve the treatment of COVID-19 infected people, and also help develop medical treatments and vaccines. The technology that we are talking about, if powered by artificial intelligence, including machine learning or image recognition and deep learning algorithms, they can be used for early detection and diagnosis of the pandemic or infection. And in addition to that, we can have more rapid drug discovery for developing new treatments. A few corporate sector companies, coupled with the Indian Council of Medical Research, they have been repurposed existing artificial intelligence systems that are initially designed for other areas to assist in social distancing, enforcement, and contact tracing. The 3D printing technology also has come in, and it can help make face masks and other personal protective equipment for healthcare workers. The swabs take less than 3 minutes to make, can be much quicker at collecting viral particles. Big data analytics is one such emerging area that also can be used to identify people that need quarantine based on their travel history, predict the COVID-19 curve, speed up the development of antiviral drugs and vaccines, and advance the understanding of the COVID-19 spread across both time and space. In countries like Taiwan, the big data analytics has been successfully applied to help identify COVID-19 cases and generate real-time alerts through analyzing clinical visits, travel history, and clinical symptoms. The infrastructure with respect to hardware and also supercomputers are required to address complex scientific problems and process big data sheets in shorter time frames in order to develop new drugs and vaccines. The COVID-19 High Performance Computing Consortium was launched to leverage the computing resources and supercomputers in the United States of America. The consortium includes 16 public and private entities such as the US Department of Energy, the IBM, and other academic and industry leaders. A similar effort was initiated in India, whereby academia, scientific institutions, private sector companies, particularly working in the pharma industry, they came together, and within less than 10 months, India was able to produce its own vaccine. Mobile technology, the applications of mobile via smartphones and via video conferencing tools can be used to track movements of individuals, alert people from visiting COVID-19 hotspots, help doctors to diagnose patients through video services, and telemedicine, telehealth support people with online shopping, electronic learning or e-learning, online meetings, and telework. Various phone and network-powered applications have been developed to help healthcare workers and ordinary people or normal people in the crisis. Robots have also been applied to fight the coronavirus outbreak. For example, the hospitals use robots as support systems to deliver food and medicine, disinfect rooms, and other hotspots without direct human contact and interaction with patients. The Internet of Things also has played a very important role. It can be used for the surveillance of people infected by the coronavirus to reduce the spread of the coronavirus. The Internet of Things also consists of several functional components, that is data collection, transfer, data analytics, and storage. The Internet of Things sensors installed on mobile phones, robots, or health monitors can be used to collect data. In addition to that, the sensor data can be sent to the cloud server for processing, analytics, and quick decision making. There are technologies, what we call it as blockchain, and this is also a distributor ledger technology that records online transactions. It is regulated through a consensus mechanism and is secured with cryptography. As an example, a smartphone application that leverages blockchain technology as well as artificial intelligence has been developed to help fight the coronavirus pandemic. The blockchain technology also enables the application to give each participant or patient a digital identity controlled by a private key that brings access to a digital version of paper, which are basically certificates issued by the government. And these certificates allow the confirmed healthy people to leave home to buy food or to go to work. So this technology is emerging now, and the blockchain has also been used to prevent the information from being manipulated by unauthorized parties. One may remember that during the outbreak, a processor which facilitated payment and financial services, a company had used a blockchain technology to monitor the process of processing claims of the people and also making payouts in a more secure and trustworthy manner. The blockchain technology has also been applied to resolve the tension and trust issues between maintaining privacy and addressing public health needs, such as tracking infected patients in the fight against COVID-19. So we have looked at biological innovations. We have also looked at technological innovations. So if you want to summarize and look at things in a broader perspective, then one can say that the COVID-19 pandemic has exposed the weaknesses of existing public health systems. If you take one example, but the use of technologies to combat the pandemic raises the challenges in many aspects. The specific nature of COVID-19 pandemic requires strong coordination of connected data, people, and systems to facilitate collaboration, not only within the nations, but worldwide. And if you want to have this collaboration, you will have a better chance to fight against the pandemic, which is coming in different forms in the recent past. Traditionally, the public health agencies and healthcare stakeholders have not used the same system, that is data formats or the systems which have been standardized or standards, hampering the ability to identify trends and develop interventions against the pandemic. The public health researchers, they also come in picture, such as epidemiologists and the government officials. They need to be connected via integrated systems with connected data to understand the evolving pandemic better and make collective decisions on addressing this particular crisis. As people play a very important role in the fight against COVID-19, it is also essential to connect, coordinate, and support various stakeholders through innovative and integrated technologies. When we talk about these aspects And when we want to summarize, then 3 to 4 points I feel should be highlighted, particularly connecting systems to integrate technologies, where the emerging technologies like IoT, big data analytics, as I mentioned already, or artificial intelligence or blockchain can be integrated to develop smart strategies for addressing immediate challenges which are caused by the coronavirus. The big data analysis of GIS or the Geographic Information System and the information of things, sensor data collected from infected patients can also assist epidemiologists to trace patient zero and help identify close contacts of the infected patients. The second point can be the connection of data to share the best practices. As the World Health Organization suggests, the new collaboration and knowledge sharing are needed to deliver targeted solutions through a coordinated effort to support countries facing stages of COVID-19 in different ways and also at different times. When we are facing the global pandemic situation, the countries need to work together to share data, to share information, to share resources, and also share effective practices and strategies to combat the coronavirus. In addition to that, the global collaboration among the relevant stakeholders between organizations and governments will be crucial to coordinating the sharing and use of data and knowledge to solve the problems we have encountered during the pandemic. Third point would be, in summary, connecting people with enhanced collaborative tools and IT infrastructure. The pandemic outbreak is rapidly changing the workplace. Millions of people are moving their workspaces to their homes through teleworking. Many industries benefit as knowledge workers learn to operate virtually, work from home, and use cloud services to process and store files. We are actually witnessing a different revolution, a virtual technology revolution, and the wider acceptance of offline or online services by people and diverse type of industries during this pandemic. The importance of IT infrastructure is enabling teleworking and online learning, as well as e-government, e-commerce, and other online activities. They have been widely recognized. The pandemic is forcing a record number of employees to work remotely, as I mentioned, for an extended duration that results heavy traffic on remote connectivity networks. In all this situation, we should not forget one very important aspect, that is to study the human behavior with technologies and digital divide. We have been using technology, but we also had our other offline activities. But because of the COVID pandemic, it is very important to study the human behavior when designing, building, and using technologies, as more COVID-19 related technologies are being developed, integrated, and used by the governments, organizations, as well as people. A lot of effort to combat the pandemic incorporate new technological advances and approaches in integrating various systems and innovations. However, we need to acknowledge that people's misbehavior with technologies may reduce the effectiveness of technology related interventions, as well as the countermeasures on containing the coronavirus break. And therefore, the information systems and technology, they will have to be attached to individual scholars, and these scholars can contribute by incorporating their understanding of human behavior into the technology design and development process, which will lead to the more effective technology. In conclusion, I would like to mention a very precise thing. When we are talking about COVID-19 pandemic, it has produced significant impact on people, business, and society. The pandemic has also implications for the design, development, and use of technology. The technologies can be useful for reducing the severity of the coronavirus pandemic's impact on people, organization, and society. However, one must not forget that the use of technologies to combat the pandemic also raises challenges such as security, privacy, biases, ethics, and the digital divide. Future pandemics are also likely to come, and while information systems and technology scholars may not be able to help with scientific aspects of developing vaccination and treatment directly, we can contribute knowledge, experiences, and time to help society better prepare for future pandemics. At this stage, information systems and technology scholars can at least help advocate and build a national common data space or health information systems for public health data sharing. Solving grand challenges facing society requires significant financial and human resources. To enhance or increase the importance and relevance of information systems and technology research, we encourage scholars, we'll have to encourage people, we'll have to encourage researchers at the university level, at the scientific institutions level, and we have apex bodies of higher education like UGC, AICTE, All India Institute of Medical Sciences, or Indian Council of Medical Research, Indian Council of Agricultural Research, where many universities, institutions are affiliated, and we will have to encourage our research students to actively apply for various government and industry grants, including various COVID-19 funding opportunities to get financial support to put some of their research ideas into practice. Since there are several small businesses in industries such as tourism, food services, and retail are being hit hardest by the pandemic, the information systems and technology faculty could collect students' biodata, put them on a Google Drive or a website, and share the resume with the interested small business owners. This will also help generate a lot of interest and enthusiasm among those who are needy or in want of jobs or some livelihood activity. This will match the information systems and technology students with interested small businesses or non-profit organizations to solve the technology and other issues they may be having during the pandemic. I'm particularly happy that some of the information systems and technology faculty in many universities are doing this and mentoring small business owners on deploying digital technologies to deal with the challenges of business community. Some academicians, some professors have known to be involved in digital solution development projects by the government of India, particularly tackling the misinformation. And they have helped organize events such as online hackathons. Many management institutions are working on that, and they're practicing to gather people with diverse skills to work on the solutions to help society fight pandemic. I believe that this will help society at large. And the example of this particular aspect, a question comes that whether the data collected from the mobile contact tracing be destroyed after the pandemic, or what kind of data management policies are required to prevent the abuse of the user data and guide the improved design, development, and use of future mobile contact tracing and tracking tools. I would conclude by saying that the biorevolutions through biological transformation, which is a confluence of advancement in biological sciences, which is decades in the making with accelerating development of computing, automation, and artificial intelligence, is fueling a new wave of innovation that could have significant impact on economies and societies, from health to agriculture to consumer goods and energy. The new capabilities and applications are already improving our responses to the global challenges from climate change to pandemic. At the time of this particular conference that is happening, I think these are being also used to respond COVID-19 pandemic. But the innovations come in, as I said, with profound risks, and we will have to remember that very often. The technology innovation also has helped enormously to resolve the issues posed by the pandemic. The challenges which are posed are resolved by experts, by the policymakers, and also by the government. But one must say that while experts have built simulation models to estimate disease trajectories that may be enough to guide decision making, to formulate policy measures, to limit the pandemic, they do not really cover the full behavioral and social complexity of societies under the pandemic crisis. Therefore, the modeling which is being done by the scientists and technocrats, they have a large potential impact upon people's lives, and that is the greater responsibility that all of us should owe. I would like to urge and call upon scientific community to improve the transparency, access, and rigor of the models that are being designed. I also call upon all the stakeholders to improve the rapidity with which the data from trusted sources are released to the community in a fully responsible manner. Responding to the pandemic is a stress test for all of us and also for our collaborative capacity and the societal, social, economic value of research. And therefore, while India is emerging with the new education policy 2020 and its implementation, it is a collective responsibility for all of us to go beyond the walls as envisaged in the NEP 2020 and collaborate, concentrate, and come out with the solutions not only for the current proper challenges posed by pandemic, but also to the future of the young generation of India. We have 18 crores of our population below the age of 18, and that is a greater challenge of educating them and making them stand on their own feet. And I believe it is a collective responsibility of all of us. I would like to thank all the organizers of this important conference with a very pragmatic and timely theme on which the event is based. And I'm sure the deliberations for the next 2 days will be not only enriching the vision of all those who are participating, but also provide a direction, a specific direction to the entire fraternity of science and technology policymakers, academicians, and educationists to take our country to a different level and benefit the society at large. Thank you very much for your kind attention. Jai Hind.
Thank you, Dr. Sanjay Deshmukh, Honorable former Vice Chancellor and Professor of Life Sciences, for your valuable speech that empowered the new ideas in the young minds gathered in this international conference. Thank you, sir.
Thank you so much.
Students remain silent. Now the session is open for queries. Students, you can ask your doubts. Thank you, sir.
Thank you very much.
Respected Vice Chancellor, sir, our students have really benefited by your talk, and we are really privileged and honored. Thank you very much, sir, for joining with us in the inauguration function, which is to be held today. So Thank you very much for your greetings on behalf of the Department of Biotechnology and Bioinformatics. We greet you and we thank you for your presence over here. And on behalf of the Department of Biotechnology, we wish you to have a bright future and also to reach the greater heights in the field of higher education to serve for the young minds in India, sir. Thank you very much, Mukesh, sir. Deshmukh,
Thank
sir.
you. Thank you, Dr. Ganesh Raja, for
Thank you,
your
sir.
commitment and your contribution to organizing this particular function. And I would like to thank the entire organizing committee
Yes,
to
sir.
have taken tremendous efforts in organizing this event. Thank you very much and congratulations.
Thank you, sir. Thank you very much.
Now let's
Kamaraj University, Madurai, and he did his postdoctoral training at Northwestern University and the University of Pennsylvania in cancer genetics. And then he joined India's leading institute, Indian Institute of Science, in 1999 as a faculty. Right now, the primary focus of his laboratory is the genetics of glioma. And glioma is brain cancer and cancer of the central nervous system. He is a highly cited author, 17,000 citations, with a H index of 49, and he has got 24 years of publication experience and several patents, as well as a wonderful research profile. And I'm sure that his talk is going to be a hallmark talk in this conference. Dear sir, I am very pleased to invite you, and without wasting much time, I want you to take over your speech, your lecture. Thank you, sir.
Unmute me. Okay, fine. Am I audible now?
Yes, sir.
Yes. Okay. Thank you. Thank you for the nice introduction, and thanks are due to Dr. Dinesh and Dr. Belba for inviting to this conference. So, audience, can you please turn on the camera to the audience for a second? Audience. Okay, sure, no problem. So, this is 1:12 right now. This is not a real correct slot of time to talk. This is actually the lunch time. Organizers asked me to talk at 1 o'clock, so I'm here. So I'm sorry if I make you wait to have your lunch, and feel free to go and grab your lunch and come back and participate. It's up to you. But organizers gave me this slot. I'm supposed to talk at 11:20. It got delayed by almost 1 hour and 40 minutes. So let me start my talk now. Share. Sorry, let's go to the last slide. Okay, can you all see the slide? Yes, can someone respond to me, please? Can you see the slide? Am I audible properly? Can somebody respond to my question? Is the slide visible? Am I audible? Okay, Okay, thanks. You all are muted, so I'm not able to hear you. So I'm assuming that you are able to hear me and you're able to see my slide.
The screen is visible now, sir.
Okay, is it in show mode? Yes?
Yes, sir.
Okay, thank you. Bye-bye. So let me start. So the title of my talk is Next Generation DNA Sequencing, the Principle and Applications. Let me start this talk with a question. Okay, why should you know about next generation DNA sequencing? And there are speakers before me and after me. They're going to talk about some important areas in the context of the pandemic challenge we are facing. Why should I come and talk about just technique? Okay, you know, many techniques are there, you know, page acrylamide gel electrophoresis, agro gel electrophoresis, isolation of DNA, you know, FPLC, mass spec, you know, so many techniques are there. The DNA sequencing is yet another technique. Why should I come and explain the principle and the applications of next generation DNA sequencing? Okay, I'll explain in the next slide how important as students of biology, I mean, there may be MSc, BSc students, research students doing PhD, and also teachers and professors. You know, if somebody knows about this technique, please bear with me. This talk is for the purpose of students who may not know how the next generation DNA sequencing works and the benefit they may get. At the end of the talk, I have given some examples. Okay, so you all know this, this particular slide. The first ever human genome sequence was actually published in the year 2001, February 2001. Two different groups have attempted to do this. One from the private company and then one from the government-sponsored project. Both of them have published in the same week in February 2001. One group published in Science and another group published in Nature. Okay, so you all know, I don't have to explain to you, sequencing the whole genome, how important it is. You all know that genes are the ones which carry out, that they code the information, carry the information. So we need to find out how many genes are there, how big is the genome. Basically, decoding the entire genome is very important to understand the whole biology of the cells. And that is true for every other organism we have in the earth. Okay, so sequencing of the whole genome without any doubt is one of the major advances we have made in our biology history. Okay, so this was done, first ever human genome sequencing was done published in February 2001. I want to take you to the next slide, how this effort was done. How long it has taken to sequence first ever human genome, number one, and how many scientists were involved, how much cost is involved, versus if you were to sequence today one human genome, how much time it will take, how many scientists are needed, how much money it would take. Okay, that's what I'm going to make you a comparison to you in order for you to appreciate that it is, why is it important to know the principle behind next generation DNA sequencing. Okay, look at here. First human genome sequence published in the year February 2001. It was the methodology used, the amount of money involved. Here you see here, the 3.2 GB back then, that is 2001. The total size of the human genome is 3.2 GB. I will explain to you in the coming slides what is the meaning of 3.2 GB. That's one cellular genome size is 3.2 GB. It has taken 10 years. The effort started in 1991 and it got completed in 2001. It took 10 years. And the total amount of money spent is 2.7 billion US dollar. If you want to know in rupee, it is about 270 crores. In other words, 27,000 lakhs of rupee was spent. Okay, and several countries worldwide participated. Probably several thousand of scientists were involved in carrying out this work, which resulted in the first ever sequence of the whole human genome. Okay, if today I were to do this, if somebody asked me to sequence a human genome today, okay, the same 3.2 GB of a human genome can be done in few days. Now the technology, since I prepared this slide, technology has improved even more. You can do it in one day, okay? And it costs around $1,000 US, which is around 70,000 rupees. If they say the cost has come down even lesser than 70,000, probably you can do it in 25 to 40,000 rupees. The cost is likely to go even down further. You look at the difference. What has taken first ever human genome sequencing 10 years sequence. You can do it in few days. It comes down to even a day or so. And then what has taken about 27 lakhs of rupees, it is only 70,000, just less than 1 lakh rupee. Okay, so that is possible. That is possible that you can do the sequencing in a very, very short time with very little money, because the next generation DNA technique, sequencing technique is available. Okay, then I would think the students would appreciate, you need to know how this technique works compared to the technique what is used for this 10-year-long sequencing that result in the first ever human genome sequencing. Look at here. How is it done? What is a major difference in principle? So the technique which is used for first ever human genome sequencing is called Sanger sequencing, which is developed by Frederick Sanger. All you do is you take the high molecular DNA from one cell, share them into small pieces, like 1 kilobase or something like that, over 1,000 bases, and then you keep sequencing. If one sequencing will take about half a day, if you want to do 2 fragments, you need 1 full day. Okay, and then there are about thousands and thousands of fragments to be coming out from 3.2 GB. If you have like hundreds of sequencing apparatus, you take about 10 years. That's what it has happened to sequence one genome. Whereas on the right side, if I use a next generation sequencing technology, I can take as many as several billions of fragments and sequence them same time. Okay, in the same time, over a period of just 1 day. Right now, I can sequence about 100 times more than one human genome is, which is 3 GB roughly. So about 100 human genomes I can sequence in 1 day today. Okay, so that means this technology, next generation sequencing technology, must be just wonderful, unbelievably different compared to this technology what is used then, that is 20 years ago. Then I'm sure the students will probably understand, they should know how this technique works. That's the reason why I chose this talk, to explain to you how is it possible to take several billions of fragments and sequence at the same time. Okay, and then get the sequence of one human genome at a very, very little cost and also very short time compared to what was taken 20 years ago. Okay, so I hope you agree with me that you would like to know the principle behind this next generation DNA sequencing. Okay, so how big is our human genome? I told you 3.2 GB, right? 3.2 GB corresponds to 23 pairs of chromosome. You all know human cells contain 22 pairs of autosomes and 1 pair of sex chromosome. All of them put together, the genome size of the human is actually 3.2 GB. To make you a little bit more understand the meaning of 3.2 GB. So you see the base, at the base, 1 base pair or 2 nucleotide is a basic unit of our DNA. DNA is a double stranded DNA, you all know. And 1,000 bases corresponds to 1 kilobase, 1,000 kilobase is 1 megabase, and 1,000 megabase is 1 GB. I told you the human genome sequence is actually 3.2 GB. Then you can understand the meaning of 3.2 GB. Okay, so you can also go by nucleotide level. 10 lakhs is 1 million, 100 million is actually 1 billion. Okay, in other words, if you come on to the left side, 3.2 GB of human genome corresponds to either 3,200 MB, 1 MB is 1,000 kilobase, please understand, or this many number of kilobases, or this many number of bases. Otherwise, you can also call it in the name of base pairs, 3.2 billion base pairs or 320 crores of base pairs. So this is the meaning of 3.2 GB. That is a total amount of the one human genome. Okay, now how was it done back then? 20 years ago, how was the sequencing done? It took 10 years to sequence. What they did is they made so-called bacterial artificial chromosome, where basically they cloned about 100 to 300 kilobase fragments, large fragments of all the human chromosomes, and then put them into E. coli. Then each of these clones were taken from a single clone of E. coli and grown in large quantities. DNA was made and then fragmented into smaller fragments. They were again cloned into another vector, common vector, and then sequencing was done on fragments on either direction. And since these fragments are made by totally by unbiased manner, and there will be overlap between the fragments, and then using the computer, you can align the sequences using the overlapping strategy. Computer will align them, give the sequence of this one particular entire back clone. Since the back clones are also made by random sharing, they will be overlaps between the back clones. So sequence of one back clone to another back clone to another back clone, by using the homology overlap between the back clones, the entire sequence of the chromosomes was established. So this has taken over 10 years with an amount of 27,000 lakh of rupees. Okay, now the three gentlemen who got the Nobel Prize for this technique, Sanger sequencing, at that time in 1980, Frederick Sanger, who discovered this Sanger sequencing, he received 1/4 of the Nobel Prize, and then Walter Gilbert also got 1/4, but he also developed yet another type of sequencing, chemical sequencing, which is also reasonably popular at that time. It is used for certain specific purposes, not for genome sequencing. And of course, the remaining half went to Paul Berg for his work related to recombinant DNA technology. So it's worth mentioning one more time, Frederick Sanger is scientist, he is no more. He passed away some 6-7 years ago, who is the one who developed Sanger sequencing. That is a technique which is used for sequencing, getting the sequence of the first ever human genome sequence. And these are the two sequencing techniques, Maxam and Gilbert, who got the Nobel Prize for the chemical sequencing, Sanger, Frederick Sanger, who got the Nobel Prize 1/4 for developing the Sanger sequencing, which uses dideoxynucleotides. These two methods are called first generation sequencing, and then the next second generation sequencing, that is what I call next generation sequencing, is called massively parallel sequencing. The meaning of that, you are sequencing large number of fragments, billions of fragments, that is massive, and parallelly same time you are sequencing. That's why the second generation sequencing is called massively parallel sequencing. It is also called next generation sequencing. That is a technique which is used right now all over the world to sequence human genomes and genomes of the other organisms as well. The third generation sequencing, which has come after the second generation sequencing, is also being used. The two major difference between the third generation and second generation sequencing is one, this is called single molecule sequencing. While in the second generation sequencing, you make hundreds and thousands of fragments, small fragments, and you amplify and sequence them. Whereas in the third generation sequencing, you simply fragment the genome and sequence each fragment without amplification. And it is also possible to sequence longer fragments, about 10 kb, 20 kb fragments can be sequenced. However, second generation sequencing will allow only small fragments like 100, 200, 300 bases. So these are the two major differences. Yet, without any doubt, the second generation sequencing, which is called next generation sequencing, came after the first generation. It's called massively parallel sequencing, has changed the way, totally the way we can sequence human genomes. Okay, so I'm going to take the next 30 minutes or so to explain how this massively parallel sequencing works. Okay, so now, this is a structure of the DNA. This is the templates DNA. There is a disturbance. Somebody is stopping and starting the recording. Please avoid disturbance, please. Okay, the organizers. So if you consider this is a template stand and this is a daughter stand, this is a growing stand. If you consider this is the primer and this is the last nucleotide which is incorporated, the new nucleotide is going to be incorporated. Nucleotide comes with the form of a triphosphate. You have 3 phosphates, and then there will be a phosphate diester bond formed. The alpha phosphate of this nucleotide joins with the OH group of the previous nucleotide which is incorporated of the ribose, and then phosphodiester bond is formed and pyrophosphate will release, and that's how the chain grows. Okay, so this is very important for you to appreciate how the Sanger sequencing works. I don't want to go into detail. However, again, I would like to show the picture of Frederick Sanger, a fantastic man who got Nobel Prize for Sanger sequencing, and also he got Nobel Prize one more time, 20 years later, for sequencing technique he developed for sequencing the proteins, and this gentleman got 2 times Nobel Prize. Okay, I don't any one of us, me or the people in the audience, will ever be able to dream of even getting a Nobel Prize. Of course, we all try to do best science possible. This gentleman got 2 times Nobel Prize. Okay, one for developing a Sanger sequencing method for DNA sequencing, and then for sequencing technique he developed for amino acid sequencing of the proteins. Okay, so this technique uses dideoxynucleotides, and these are molecules that resemble the normal nucleotide, that is nucleotide triphosphates, but it has, instead of OH group, you can see a normal nucleotide at the third carbon, you have this OH group. This is a normal deoxynucleotide triphosphate, whereas the dideoxynucleotide has this OH group replaced by hydroxyl group. Whenever this dideoxynucleotide gets incorporated, the polymerization will stop, and then wherever it stops, the length of the oligonucleotide can be identified by running a gel and through autoradiography. So this is a simple description of how Sanger's dideoxy sequencing works. If you consider this is a template, you have the primer added here. Primer is labeled at the 5' end with a phosphate, and then you divide them into 4 tubes, and then reaction going to start by adding DNA polymerase and the 4 deoxynucleotides. Okay, these are triphosphates, and then you also add one of the nucleotides as a dideoxynucleotides, DDGTP, DDATP, DDCTP, and DDTTP. So what will happen is when the polymerization stops, the normal nucleotides will start the polymerization. And the DDoxynucleotide, which one of them is added to each of the tube, will go and stop at different positions. So in that case, in each of the tubes, wherever the polymerization stops, it will result in the varying length of oligonucleotides generated. Wherever it stops with the G here, A here, and C here, and T here. And you can run all these 4 reactions in 4 lanes and do autoradiography. This is how the ladder looks like. By reading the sequence from the bottom, you can get the complete sequence of the fragment to be sequenced. Later on, it went to the fluorescent platform where you can, each of the dideoxynucleotides are simultaneously, labeled with a different flow chrom. So you don't, instead of running 4 lanes, you can run 1 lane and then get the sequence. And the technology has improved that you can do it much faster and you can get a much longer reads like 1 kilobase. Okay, that is what used for sequencing the first ever genome draft. Okay, now I'm going to compare the technique, how it works in the so-called Sanger sequencing, which was used for the first ever human genome draft versus the next generation sequencing technique. Just to compare, make you understand how much it is different, and then explain to you more in detail how on the right side next generation sequencing works. So in the left side, which is called capillary sample preparation, that is a traditional Sanger sequencing, you have a back clone, you divide them into, share them into small pieces. Each of the small pieces cloned into a plasmid vector, and then put it into E. coli, make large amounts, and then use this as a template to do sequencing. Look at the lot of effort you have to put in multiple steps. On the right side, same large fragment, if you consider, share them into small pieces and put each of these pieces attached to the surface of a glass slide, same microscope slide, what we are talking about, what you use in a microbiology class. And then each one of the fragment you amplify on this glass slide to make multiple copies of the same fragment and then use it to sequencing. Look at here in the very first itself, so many steps are avoided. You don't have to clone the fragments and you don't have to make the DNA again. Here, you just simply share the large fragment into small fragment as much as what you do here without any need for cloning and making individual fragment plasmids. You directly attach to the surface of the microscope slide and then amplify in situ on the slide and then sequence it and get the sequence information. Okay, so many steps are avoided here. That's why you are able to sequence several thousands of fragments simultaneously. Now, how does the sequencing work? In the case of capillary sequencing, so-called Sanger sequencing, you have the template, and this yellow portion is actually the primer. Primer binds, and then this polymerization occurs. It stops at different nucleotides based on the dideoxynucleotide which get incorporated, and the products are ran on the gel, and the products go like that. When they reach the end, laser is passed out here and identifies which of the dideoxynucleotide is incorporated, thereby it will tell you the sequence information of the particular template. Whereas here, instead of laser running a gel in the form of capillary, what you do here, you are using a method called sequencing by synthesis, wherein what you do, consider this particular fragment is the one, this particular fragment is equivalent to a fragment sitting here on the surface of the slide. Okay, so what I do, I add a primer, okay, and then start the reaction. I have 4 deoxynucleotides, and each of them are dideoxynucleotides. So only one nucleotide will be incorporated, and then it cannot perform polymerization further because the 3' hydroxyl end is not available. It is protected. It's kind of equivalent to dideoxynucleotide. It stops after the incorporation of the first nucleotide. It could be any one of them, G, A, T, or C. Then what you do, you have a camera which goes around, captures the image of this particular fragment. Okay, whatever the color which is there, the image will tell you for this particular fragment in the cycle 1, that is a nucleotide, first nucleotide to be incorporated for a particular fragment. Remember, this fragment is permanently attached to the surface. Okay, so for this particular fragment in the first cycle, one particular nucleotide carrying the green nucleotide is incorporated. Then what you do, you reverse the termination. you make sure you do some gimmicks, chemical gimmicks to remove the 3' hydroxyl end protection, and then add again 4 nucleotides. Each of them are dideoxynucleotide, kind of dideoxynucleotide, that is hydroxyl end is protected, carry different fluorochromes. Now yet another nucleotide will be incorporated. Let's say in this case, instead of green first nucleotide, second nucleotide is blue nucleotide. Okay. Now again the camera comes, captures the second cycle nucleotide which is incorporated. So the first cycle is green, that is the first nucleotide. Second cycle is blue, that means second nucleotide is the blue nucleotide, and go on, third, fourth, fifth, and finally the image will compare, the program will compare the image taken for a given position, first cycle, second cycle, third cycle. Based on the color, the image will stitch together, the sequence of the particular fragment will be given. Okay, so this is how the technique works. So in the case of capillary, you have one tube, one template, one capillary, you get only 1000 bases at a time. Whereas in this next generation sequencing, you have one chip containing millions of fragments, you get gigabase of sequences. So you can understand the difference between how much different the next generation sequencing compared to the capillary sequencing, so-called Sanger sequencing. So next generation sequencing is here. So let me go to the next slide. So, so there are 3 next generation sequencing technologies developed simultaneously about 15 years ago for whatever reasons, so-called Solaxa sequencing, which is based on the reversible dye termination, which I explained to you a few minutes ago. This appears to be the most popular. This is actually sold by, this technology is sold by the company Illumina. They claim world's 95% of the sequencing is carried out by Solaxa sequencing. So I would think the students would like to know, how does the Solaxa sequencing work? This is the technique which is used to do sequencing today. About more than 95% of the all sequencing all over the world, this is a technique being used. So that's the reason why I chose to explain to you how the Solaxa sequencing, which works based on the reversible dye termination, works. Okay, so at this juncture, I would like to bring this gentleman's name, Shankar Balasubramaniam. He is one of the scientists, along with David Klernerman from Cambridge University, actually responsible for discovering this particular technology. Okay, I think it's very important to tell, is one gentleman from Tamil Nadu who speaks Tamil, a scientist involved in discovering this particular technology. Okay, this is the complete principle of how this Illumina Sanger sequencing, Illumina based reversible dye termination sequencing works. Again, basic principle, look at this, is a template DNA, and the blue line is actually the primer, and the RNA polymerase, sorry, the DNA polymerase is sitting here. It is ready to incorporate the nucleotide. Whatever is there in the template, the complementary nucleotide will incorporate. You all know that, right? So you have 4 nucleotides, which are each one of them, GATC, is labeled with different fluorochrome, and each of them have a 3' hydroxyl end protected in such a way, one more nucleotide cannot be incorporated to such a nucleotide. And then you need to deprotect the 3' hydroxyl, make it available for next cycle to move on. So what happens? One of the nucleotides will be incorporated. Here it is A in the template. That means automatically T will be incorporated. Once the T is incorporated, T carries a green color fluorophore. So the laser is excitation is given. The camera comes and takes the image. It will make the image in such a way that this first nucleotide is green color nucleotide, and then remove the fluorophore and remove the blockage. 3' hydroxyl end has to be made available for the next nucleotide to be incorporated. And here in the template, next nucleotide is C, and so the G will be incorporated. We keep repeating this cycle. For every one cycle, one nucleotide will be incorporated, and the information which nucleotide is incorporated is stored by the image for a given position, which is represented by one particular fragment, which always remains in the same place. And then the program will stitch the color of the nucleotide, incorporate in the first cycle, second cycle, third cycle, fourth cycle for the given fragment, and put them together, and the program will tell you what is the sequence of the template in the form of a newly synthesized oligonucleotide on the top strand. This is the simple principle by which this technique works. So let me take you through very quickly, little bit more detail, how it actually works. It is not possible, so easy. It's not so easy to attach a DNA fragment onto the surface of the glass slide. Please understand, it's not easy to attach permanently onto the glass surface. So consider these the gray color double-stranded fragments are randomly shared fragments coming from a high molecular DNA, let's say coming from human cells. You want to sequence each of these fragments. You want to attach each of these fragments onto the surface of the glass slide. Then you want to do sequencing the way I explained to you just few slides ago. So what you do to attach the fragments onto the surface, you need to attach something called adapters. There are two kinds of adapters, just for make it easy for you to understand. There is a purple adapter, there is a pink adapter. You attach them in such a way, one goes to on either side. Okay, the reason why you are attaching the adapter is coming to the next stage. So this consider where is my mouse? Okay, so consider this is a microscopic slide. In the microscopic slide, there are something called lawn primers. Primers are nothing but single stranded oligonucleotides which are permanently covalently attached to the surface of the microscope slide. These are single stranded oligonucleotides. The sequence of these oligonucleotide primers are complementary to these adapters, which are actually attached to the randomly shared chromosomal DNA fragments. Now, when you add this adapter ligated DNA fragment onto the microscopic slide, what will happen is by using the complementarity between the adapter sequence and the lawn primer sequence, the adapter ligated fragments will go unattached onto the surface of the glass slide, something like this. And then once it is attached, you can use something called bridge amplification, a modified form of PCR. I will explain how it works very easily. You can understand as we go along. You actually end up making large number of fragments, same copies of the same DNA fragment like this. So this is what called one cluster. Cluster is nothing but, you see here, this is a microscope slide. On this lawn primers are there, two types of primers are there, pink primer and then the purple primer. And the adapter ligated library fragment come and join as a single fragment using the complementarity between the adapter and the primer. And then using the bridge amplification, you do a localized PCR reaction and make multiple copies of the same fragment. So this is called one cluster. Cluster is nothing but about 1,000 copies of the same DNA fragment is copied several times. Now we are ready to go for sequencing. Okay, so this I will skip it. I will come back to this very quickly. So the multiple steps involved in sequencing is you prepare the sample and then you prepare the clusters, which is multiple copies of the same DNA fragment, and then you go for sequencing, and there is a lot of data analysis involved to get the sequencing of the particular genome. Okay, library preparation. How do you prepare the library? I told you very simple manner few slides ago. You fragment the DNA and then repair the ends, because when you fragment, ends will be of different types. You know, blunt ends, 3' protruding, 5' protruding. You put a cocktail of enzyme and make them as a blunt end. Okay, and then ligate the adapters and then select certain small size of the fragment and load onto the particular slide to develop the so-called the clusters. So when you fragment the DNA, when you fragment the DNA, ends will be of different type. Consider this is one large fragment coming from the chromosome. Ends will be of different type of ends. They may not be all of them are the perfect ends. So you put a cocktail of enzyme to repair them. Okay. So this is the repair occurs and both sides become blunt. Okay. Now in order to ligate the adapter, what you do, you put a extra A. Okay, extra A is the 3' hydroxyl end. 3' end, you put an extra A because adapters come with the T overhang. Okay, using the T overhang adapters, you ligate to the A overhang fragments. That's how the adapter is ligated. Okay, so consider one side is a pink adapter, consider another side is a purple adapter. Now we are ready to go into the microscope slide. Let's see how does it look like. So this is a microscope slide. The ligated DNA, adapter ligated DNA is added on to the microscope slide. Now let's see how does it work. Let me skip this slide. Okay, so now look at this here. Next step. Okay, this is a real glass slide where the sequencing occurs. Okay, so now let's look at, this is a surface where the lawn primers are there. Two types of color primers are there. Sorry for changing the color. You can see one is a yellow, another one is a blue color. Okay, so one of the adapter ligated fragments, single DNA fragment, single DNA or single stranded DNA binds to the blue color adapter binds to the blue color lawn primer using the complementarity. Please understand the primer, lawn primer is attached covalently to the surface, whereas the DNA fragment single strand is not covalently attached. It is attached to the lawn primer by using the complementarity. So the fragment is not attached permanently to the slide. Please understand. Now you change the condition in the slide in such a way. Sorry. So what you do next step? So this long primer is having a 3' hydroxyl end, which is the one which can be used to grow the nucleotide strand. You add the polymerase and nucleotides. Now you can actually copy the stand opposite, complementary to the original stand which gone in. Okay, so we copied the complementary stand, complementary sequence is copied. We don't need the original stand anymore. So you can get rid of it. Okay, we got the complementary sequence. So we remove the original stand. Now you want to make we want to copy this, make more copies of the stand. What you do, you modify the condition in such a way, you bent it so that the other end of the DNA binds to the orange color primer, and the orange color long primer will have 3' hydroxyl end. So now you actually copy it now, other way around. Okay, and then make the equivalent of the stand which is thrown out, original stand. So you repeat this process multiple times. Finally, you will have several copies of the original DNA fragment made. That is what called one cluster, which I showed you few slides ago. So now we have a cluster ready. Cluster is nothing but multiple copies of the same DNA fragments. We are ready to go for sequencing. However, in order for you to sequencing, we need only one template. We cannot have both strands. So remove one of the strands. Okay, some chemistry is used to get rid of one of the strands, keep only one strand. So consider there are 1,000 copies of the same strand sitting here. For easy observation, understanding, I put only 2 strands of the same fragment. Now we are ready to go for sequencing. What you do, they just make sure that all the free ends are blocked, and then you add a primer. Okay, primer is added. Now the primer is ready to start the sequencing. Okay, now what is the next step? All the 4 3' hydroxyl end protected dideoxy, equivalent to dideoxy nucleotide, but each one of them labeled with a different fluorochrome, right? All of them are added. Only one nucleotide will be incorporated because they all have 3' hydroxyl end protected. So it so happens, so-called the dark reddish color nucleotide is incorporated. Pass the laser and take the image through the camera. Okay. Now what you do next step? Remove the fluorophore, unblock 3' hydroxyl end. It is ready to take the next nucleotide. So, and repeat the same protocol again. Continue to do each cycle, one nucleotide is incorporated. Image is taken. Okay. And then stored. Cycle 1, cycle 2, cycle 3, cycle 4. The position of the cluster remains in the same place. So at the end of it, the image taken at the first cycle will have thousands and thousands of spots. Each spots corresponds to one strand, one position, one cluster, information for one particular nucleotide in the form of one color. So in the same position, image after image, the color of the nucleotide is stitched together to get the sequence of the particular fragment. This is how the result looks like. A small portion of the image where about some 20 different clusters, 20 different fragments are sitting there, and cycle number 1, cycle number 2, 3, 4, 5, 6, 7, 8, the images are shown. You can see here in all these images, the position of the spot remains there. It does not change. Only the color changes depending upon what nucleotide is incorporated in that particular cycle. If we take the 2 examples, example spot number 1, cluster number 1, fragment number 1, you can call it either any way you want. First nucleotide is T, then it is G, C, T, A, C, T, G, A, T. Same is true for the other spot or other cluster or other fragment. It so happened multiple T's are incorporated one after other. That is because it has 7 T's, T7 followed by G and then T. This is how the next generation sequencing based on Solaxa sequencing, so-called reversible dye-termination methodology works. I hope you understood. So I'm going to take you through next 5 or 6 slides to give you some example how this can be applied to, including for SARS-CoV-2 research. Okay, so once you do that, you get the sequence of individual fragments. You have billions of fragments. Okay, you have billions of small size fragment sequences sitting in a pot. How do you put them together and get the sequence of the entire chromosome? Remember, the very first step, you take the large high molecular DNA, shear them into small pieces randomly. When you shear them randomly, there will always be chances that next neighboring fragments will have an overlap with each other because sharing happens randomly. Okay, so you put all the sequences into the computer. A computer program will identify by using the partial homology between the fragments, overlapping fragments. It will stitch them together to get the complete sequence. This is all the principle by which next generation sequencing works. I almost come to the end of it. I already explained to you right now how the next generation sequencing works. As against original Sanger DNA sequencing, you take only one fragment, set up one reaction, whole day you sequence to get the 1 kb fragment sequence. Whereas here, you take billions of 1 kb fragments or 500 base fragments. Sequence same time in one glass slide. Program will stitch them together to get the sequence of the entire human genome. I hope students, you understood how the next generation sequencing works compared to the so-called Sanger sequencing, which was used originally to sequence first ever human genome, which took about 10 years compared to this technique, which will take right now less than a day to sequence the whole genome of the human. Okay, now once the sequence information of one fragment is called a read, you align them to the human genome and then get the sequence of the new genome, what you are going to study. So this is how the reads aligned to the genome look like. And again, one more picture. You can also find out if there are mutations. One of the applications of next generation sequencing is you can sequence the cancer genome coming from the cancer tissue. Are there any mutations in the genome or DNA of the cancer compared to the normal tissue? Okay, all you have to find out, most of the reads, the sequence coming from the different fragments will exactly match to the so-called normal genome. If there is any mismatch between the cancer genome fragments compared to the normal genome fragments, it tells you there is a mutation there. That's what I have shown in this picture. Okay, nothing, very simple. If there is a difference in the sequence, nucleotide sequence at any place coming from the reads, coming from the fragment information from the cancer genome compared to the normal genome, it indicates that there is a mutation. Very simple. Okay, so I'll skip this slide. So the variety of applications are for next generation sequence. You can sequence the whole genome. You can sequence only the exome. Exome corresponds to coding region of the genome. For example, in the human, the coding region which codes for protein is covered by only about 2% of the whole genome. So only 2% of genome carries the information for protein coding sequences. So you can only sequence that portion because they are the most important region of the genome. You can sequence the RNA. Those days when you want to do transcriptome changes in the RNA level between the two conditions, you used to do microarray, right? Instead of microarray, you can actually sequence RNA. The principle is the same. You take the RNA, share them into small pieces, convert it to CD and double-stranded DNA, then you adopt a ligate, put it on the glass slide and sequence it. Principle exactly same. You start with the RNA, convert to DNA, and then ligate the adapter, put it on the microscope slide, do the sequencing. Principle exactly remains same, except a small change at the beginning of the sample preparation. You can sequence the mRNA, purified mRNA as against a total RNA. You can sequence small RNA, microRNA, which are small RNAs. You can do chip sequencing wherever on the DNA bound, protein bound DNA fragments. If you are working on a transcription factor, you want to know where does it bind onto the chromatin, onto the DNA. You can immunoprecipitate using antibody against a protein which binds a specific part of the DNA, pull down the DNA fragment, sequence it. That's called RIP sequencing, RNA immunoprecipitation sequencing. If there are certain RNA to which certain protein binds, the RNA which are specifically bound to a protein can also be sequenced. Deep sequencing, if there are certain mutations occurs at a very, very, very low percentage, you can do sequencing at a higher depth. That's called deep sequencing. You can do amplicon sequencing. You don't want to sequence the entire genome. Similar to whole exome, even whole exome I don't want. I want to sequence only 100 genes. 200 genes. You can sequence only those set of genes by doing amplification of that particular area and then sequence it. Targeted sequencing, similar to amplicon sequencing. Methylation sequencing, you know, the promoter gets methylated in the CpG island that leads to repression of gene expression. That can also be sequenced and identified where the genome methylation is occurring. There is also another form of sequencing called attack sequencing, assay for transpose accessible chromatin. You know, the chromatin, certain regions are opened up, that is amenable for activating transcription, certain promoters are actually closed, which are repressed. So which regions are actually undergoing active transcription, that can be identified using this assay. De novo sequencing, if there is a new genome, suppose you are finding a new bacteria which is not known before, sequence of the genome is not known, you can sequence and find out first time ever, you can establish the sequence of a new bacteria what you isolated. That's called de novo sequencing. 16S metagenomic sequencing, often if you want to find out the particular species in a particular genus of an organism, you can use 16S RNA, which is having a highly conserved genome sequencing, right? By comparing that, you can find out which particular strain, which particular genus they belong to. So you can do that kind of sequence. So variety of applications are there for next generation sequencing. What about the third generation sequencing? I told you in the beginning, the difference between the second generation is called next generation. The third generation is called small molecule sequencing, single molecule sequencing. You can sequence only, take one fragment, no amplification, sequence just simply directly. And you can sequence large fragments, about 10 kilobase and 20 kilobase. There are two technologies which are actually fast coming. A lot of people are using. I don't want to go into details. One is Pacific Biosensor, which is called the SMRT sequencing, Oxford nanopore. These are the two besides the another technique which I have shown here. I will not go into details. Somebody can read it. I will share the slide to Dinesh. Anybody wants to read more about it can actually read and understand. So now coming to the last 5 slides, what about pandemic? What about SARS-CoV-2? It's a next generation sequencing helped in any way to understand the SARS-CoV-2, the pandemic situation. You all know the SARS-CoV-2 virus, how it looks like. The spikes which are there on the top, brown color, they are the ones which are formed of S protein of SARS-CoV-2 virus. Okay, so if you look at here, this is a genome of the SARS-CoV-2 virus. This is about 29,000 bases. Okay, and there are many proteins encoded by this, and you have a very long protein. This is called RNA dependent RNA polymerase, which is very important for replication of this virus. There is something called spike S protein, which is very important. This is a protein that looks like as a spike. This is what equivalent to this red color spikes what you see on the left side. And there are many other proteins. N protein, say envelope protein, which is also there here. So there are 2 things I want to know. All of you know, students and even the people, common people who are all walks of the life, if you go to your home, your parents, everyone knows the word RT-PCR. RT-PCR is a technique to diagnose whether the person is infected with the SARS-CoV-2 or not. What do they do? You all know RT-PCR. You basically take the RNA, convert it to cDNA, do PCR to find out whether a particular sequence is there or not. So all you do here is to see whether a given person is infected with SARS-CoV-2, you collect a small sample from your nose and then make RNA out of that. Considering there is a virus, you can lyse it and make the RNA, convert it to DNA and do a PCR. Not one PCR, do 3 PCR. One region specific to this first gene, and then another one specific to S gene, another one specific to N gene. All 3 genes, 3 amplicons, 3 PCRs, all of them should show positivity in order to say a person is positive for SARS-CoV-2. This is the simple principle of RT-PCR. Now, where does the genome sequencing comes into picture? RT-PCR is not genome sequencing. You are only looking for a presence of particular region of the virus, 3 different regions corresponding to 3 different genes. Whether it is present or not, by doing RT-PCR, you are confirming a person is positive or negative for SARS-CoV-2. Okay, if the person is positive, then you have to take this virus and sequence the whole genome. Why do you have to do sequencing? Then only you will come to know whether the virus, so-called, you remember 2020, April, first ever lockdown started. Okay, and then at that time, so-called G virus, G-clad virus was predominant. Later on, the second wave, people said there is a Delta virus. Okay, and now they call it as a Omicron virus. What are these? What is the difference? They are all one and the same SARS-CoV-2, but there are variations in the sequence of the genome, and that variations cannot be identified by the RT-PCR. One has to take the entire RNA, sequence the entire RNA, then only you can find out that the particular variant is there or not in our population. Okay, so now this is the Omicron virus. This particular virus compared to the original virus carry about 60 mutations. Okay, out of them, you can see here the S protein is shown, but it's not drawn to the scale. The S protein, which is not that big of the entire virus, is shown in occupying a large proportion of the virus, carry about 32 mutations of the total 60 mutations. Okay. That's why it's called Omicron. It seems to spread much faster, even though it doesn't cause a much aggressive disease. Okay, so in order to find out these kind of changes, whether it is a G virus, Delta virus, or Omicron virus, it is important to sequence the entire genome of the virus. So if we consider this is the original Wuhan virus, it is about 29,000 bases. You have to make multiple amplicons, like the 3 RT-PCR done for the identification of the virus. You do as many as 20 to 40 amplicons, 20 to 40 RT-PCR, and then sequence. Remember, these fragments are overlapping to each other. So the after sequencing, getting the information or sequence of each of these fragments is stitched together by the program to get the complete sequence of the SARS-CoV-2 genome. Then by comparing the sequence of the particular virus, which is collected from an individual, comparing that with the original Wuhan strain, you can find out what kind of changes are there in the new viral genome, and then thereby you actually record whether it was the original G virus or Delta virus or the so-called Omicron, which is a recent variant. Okay, I hope you understand how the next generation sequencing helps us to identify whether at this time Omicron is present or not, and what proportion Omicron is present. This is one of our contributions early on in April, May, June 2021, when the virus just started spreading into India. I'm talking about going back 2 years. Okay, April, May, June 2020. Okay, our country is one of those countries who stopped international flights much later by the time virus started coming to India. From where it is coming, okay, we don't know. Even though virus started from China and it went to different countries, came to India, because we don't have too many people going to China, right? So, but the virus has come. It must have come by travelers from China to some other country. From there, somebody would have come to India. Most likely that should be the route of the spread of the virus to India. We did an early on and work which found out that the spread of the virus looks like the Indian possible origin to be countries mainly from Oceania, that is Australia, Europe, Middle East, and South Asia. This is one of the papers we published long ago in bioarchives, and also we published that in Current Science a few months later. Okay, now the last 2 slides, Dinesh, I have a couple of more minutes. I started around 1:15, only 2 minutes I'll finish. Is it okay, Dinesh? Dinesh? Organizers? Okay, 2 more minutes. Okay, we also carried out next generation sequencing. As in the introduction, someone told to the audience, my lab works on genetics of glioma. Glioma is actually the one type of very commonly occurring brain tumor. We wanted to know what kind of genetic changes are occurring in the genome of the Indian glioma patients. So we basically sequenced several numbers of glioma tumor samples and also matched peripheral blood for reasons to get rid of those individual specific variations. And then we identified several mutations. I wanted to show you one particular finding. We found out mutations in several particular pathways, they are inactivated, that results in a very aggressive tumor. And this is how it looks like. One particular pathway called neuroactive ligand receptor interaction pathway. Whenever members of this particular pathway is mutated, and we found those patients having mutation in this pathway, the virus, the tumor is highly aggressive, the survival of the patient is very, very lesser. Okay, what is this particular pathway? This particular pathway contains about 750 GPCRs. There is G protein coupled receptor. These are 7 transmembrane protein membrane receptors which are located on the membrane. There is a N-terminus outside and C-terminus inside. N-terminus, whenever the ligand binds, and there is an activation of, and then signaling goes down, and that's how the G protein GPCR signaling works. You can see the Kaplan-Meier survival curve. The red line corresponds to those patients who have defect in the GPCR pathway. You see the median survival comes down to almost 3.148 months. Whereas if the pathway is wild type, if the pathway is not affected, it's completely wild type, the patient's tumor is less aggressive, they live longer, 14 months. You can see the difference, 3 to 14 months, about 5-6 times longer in terms of median survival. Then you can appreciate defect in this particular pathway makes the tumor highly aggressive and results in the lesser median survival. So one of the GPCRs, which is very commonly mutated out of this 750 GPCRs called CalCR, calcitonin receptor, we took for additional investigation and found out there are 7 mutations. Any one of these mutations, this receptor will make the tumor highly aggressive. One of my students spent a lot of time, about 2 years. You can see again, CalCR mutated patients have a lesser survival compared to those who are wild type. Okay, so she spent a lot of time to find out the CalCR in the patients where it is wild type, when there is no mutation. CalCR, when it is bound by the calcitonin, which is a ligand for it, it activates and then inhibits certain pathways. When it gets activated, it inhibits certain oncogenic pathways, resulting in less aggressive tumor. So if the receptor is CalCR, GPCR is wild type, it acts as a tumor suppressor, growth inhibitory protein, which means it's good for the person that the tumor is less aggressive, they live longer. On the other hand, in those patients where it is mutated, this function of this protein is affected. Calcitonin cannot activate this GPCR, which means the oncogenic proteins which are inhibited on the left side are not inhibited, so the tumor becomes highly aggressive, resulting in the worst survival. This is our major finding. Based on that, we are trying to develop some therapeutic aspect. That is, wherever the tumor which is wild type CalCR, we can activate the CalCR by adding calcitonin, thereby make the tumor less aggressive. So that can be added to the current therapy, thereby one can increase the median survival. You can see that is the approach. Calcitonin is used for osteoporosis, particularly for post-menopausal women. It will increase the bone strength. We decided whether you can use calcitonin for inhibiting the growth of the brain tumor. You can see here the brain tumor cells are grown in the flank, not in the brain, of course. And you can see on the left side, the animals received PSA as a control. The tumor continued to grow. It is actually shown as a bioluminescence because these cells are tagged with luciferase. Whereas on the right side, the animals which received a calcitonin, tumor initially was there and then completely disappears, suggesting that calcitonin treatment can be used to inhibit the growth of the brain tumors. So the idea is that right now, the current treatment is actually surgery, irradiation, temozolomide chemotherapy. The median survival is only 15 months. If we can add calcitonin to this treatment, if we think that it can increase the median survival 20 or more months, that's the kind of benefit we think we will get by using this strategy. Okay, I'll stop here. Acknowledgements. This work, the last part of the work which we carried out on CalCR is carried out by Jagrati Pal and Vikas Patil. These students already left. They are doing postdoc elsewhere. And Mainak Mandal and Ankita did the work related to the SARS-CoV-2 which I presented. And my collaborators on the right side, funding to various agencies. And I'll stop here. If there are any questions, please ask me. I'm done. 2:05. I think I took about 55 minutes. Audience, organizers, are you there? Hello. My talk is over. Dinesh, there are 2 hands raised up. Ramya, Kiran Mai. Is there any organizers to come to moderate the question and answer session?
Yes, sir. Yes, sir.
Okay. Yes. Great.
I request those who are attending in online, you can interact with our Professor Kumaravel sir, who is the person who is working in the Indian Institute of Science. Kindly ask, madam.
Hi, sir. Am I audible?
Yes, yes, please go ahead with the question.
Thank you so much, sir. It is very beautiful and nice presentation. I just acquire so much of knowledge about this.
Thank you.
And whatever this confirmation of virus, why don't we use identification of spike protein confirmation for the viral infection?
What do you mean by confirmation? I didn't understand.
I mean, whatever these nowadays, the fragments of viral DNA, I mean, RNA that is converted to cDNA, as well as it is going to identify whether the viral infection is present or not. Whatever, through that way, if we want to shift to the protein identification instead of that, spike proteins are there, isn't it?
I totally agree with you, madam. At the end of the day, it is a protein which gets changed, and that's why the virus infects faster than the old virus. There is no question about that. But then the information comes from the DNA. It's not easy to take a protein and sequence a protein. Okay, so that's why you have to take the genome. And one thing I can understand from your question, instead of sequencing the entire genome, the most of the important variation, whether it is a G virus originally or Delta virus and Omicron, they are all in the S protein. Instead of sequencing the whole genome, you can sequence in the S protein alone. That is fine. I agree with it. However, most of the genome studies involves the entire genome because you also want to make sure there are no other changes occurring elsewhere in the part of the genome as well. That is why usually genome sequencing involves the entire genome. It doesn't really take much time. You can just do it in a couple of hours to sequencing one genome at a cost of less than 10,000 rupees today. Okay. Yes. Very, very cheap.
Sir, I had another. Yeah.
Yes.
Thank you so much, sir. And I had another doubt. If there any method, is there any method to silence the S protein?
Why do you want to silence S protein? Answer me.
Because it is infecting the human cells, isn't it, sir? Through the S protein.
Correct, madam. But S protein is not in our body. S protein is part of the virus, right?
Yeah.
See, we don't know how to control the virus. We cannot control the virus. Only thing is we can protect ourselves wearing a mask, maintaining a social distance. There is no way you can control the virus. We can only prevent the virus infecting us, right?
Okay. And what is about the silencing of viral protein after infection, sir?
After infection, I'm not sure whether you can inhibit the viral protein after the infection. Only way is to, you can take all this, natural precautions to immune status, increase the immune status and all those things. You can get yourself vaccinated. So the immunity will prevent more virus setting up. The disease can be inhibited. So that is the way you can inhibit the virus. Okay. So it is an example. Vaccination has helped us from the third wave. But for the vaccination, Omicron third wave would have been a disaster as good as the second wave. You all have witnessed to that.
Yes. And what may be the cause for the mutation in virus, sir?
It's very simple, madam. It is very simple. Mutation occurs due to error carried out by the polymerase. Number one. Polymerase does the replication of the viral genome, right? When it adds the nucleotides, it will do some mistake. That is what results in the mutation. Number one.
Yeah.
It so happens the polymerase which is sitting in the virus is RNA dependent RNA polymerase, which is a very, very bad polymerase. Its proofreading capacity is very, very bad. More mutations are incorporated. Number two. Number three, more the virus infects, more the chance of new variants emerging. That's how it occurs.
Oh, okay, sir. Thank you. Thank you so much.
Thank you.
And I think, what are the causes? What are the factors that are causing the mutation of this viral DNA?
No, that's what I'm saying. No, more the see. If you put mask, keep a social distancing, you are not letting the virus to infect. Okay, that means virus is not allowed to replicate. Where does virus replicate? Only when it infects a human being. If all of us are so careful, don't allow the virus to spread. Okay, if in a small village there is only one person infected. Okay, you take that person, isolate them, keep them in one home. All other people don't go to the person. So you are not allowing the virus to spread to the other people, and you are not allowing the virus to replicate. So when the virus is not allowed to replicate, it doesn't get a chance to develop the mutation. So preventing the spread of the virus is the one biggest method by which you can prevent new variants from occurring.
Okay, what is about the hand alcohol used in, I mean, whatever the chemicals that are used to prevent the viral transmission?
You are talking about sanitizers. They are very, very important.
Sanitizers. If they are giving, I mean, okay. If these sanitizers are causing some mutation in viral genome?
No. They are used to, suppose when you are using your hand, you touch this bottle, somebody would have taken the same bottle for drinking water, or a pen. For example, somebody used a pen, I borrowed a pen from someone, and they will be touching their nose, and then virus will spread through their hands. So if you use a sanitizer very often, what happens? Sanitizer will kill the virus. Virus is made up of proteinaceous coat. Okay. The detergent will just kill the virus. That is the purpose. So using a detergent, sanitizers are very, very good. Continue to follow. Omicron might have gone, appear to have gone, but it may come back again. It may come back again. We thought at the end of first wave, everything is gone. Suddenly second wave came. And then we thought second wave, it's over. Now third wave come. Fourth wave may come. I have no idea. But let us be very cautious. Wear mask, sanitize their use, keep social distance. Okay, don't go to restaurants and huge crowd, cinema theaters, unless and otherwise it is essential. Still wear a mask.
Okay. Sir, at the end, you had explained
I have a
the
doubt.
tumor. Hello?
Yes.
Yeah, tumor development and explain about the calcia protein, which is preventing the tumor formation in treated mouse, isn't it, sir? If this uptake of more protein, which maybe prevent the tumor formation?
I don't know whether it will prevent the tumor formation. If there is a tumor, if you treat the patient in the mouse model we show with the calcitonin, it will inhibit the growth. That's all we know. Whether it will prevent the tumor formation to begin with, I have no idea. Okay. Fine.
Okay.
There is some student wanted to ask a question. Madam.
Thank you, sir. Thank you so much.
Thank you very much. Some student, Ramya has raised her hand. Rakesh has raised her hand.
It was a very good lecture, sir. I have enjoyed your lecture. I'm working on the sequencing only for the cyanobacterial culture. The thing is, I have sequenced the three cyanobacteria totally, where one I have got the novelty work, but it was not matching with the NCBI. The peak and all I have got very clearly. I have one doubt in that. One in a forward primer, I have got 50,050 amplification. It was amplicon. And in another reverse primer, I have got 15,250 bases. So what is the, why that that much difference between the forward and primer after get the results from sequencing, sir?
This is very specific for a sequencing project. I am not a person to answer your question. You need to talk to a sequencing expert and go with the data, sit with them. You also need someone who is called data analysis person, which is again a very important skill, which none of the scientists like me and the regular scientists like Dinesh may have unless he knows. The data analysis is quite skill driven. Those who are doing computer programming will only know. So you need to discuss with the people who are actually doing sequence and find out what is your problem. I really don't have an answer for your question.
Okay.
I think if in principle, if I can understand, you are trying to sequence a new genome, so-called de novo genome, which is quite tough. You need to take some expert advice. Thank you.
Okay, sir. Thank you, sir.
Ramya?
Sir,
Ramya has already asked a question. Rakesh? And anyone else? Organizers?
Sorry for the inconvenience, sir. Next speaker, Dr. Jayakanthan sir from Malagappa University is waiting. So if you have any queries, sir, mail, we will give you the mail ID of the...
Sure. Please send emails to me. Bye-bye. See you all.
Yes, sir. Sir, thank you very much, sir. Thank you.
Now let's move on to the next plenary lecture. I invite Dr. Sherlyn Rosita, Assistant Professor, to give a brief introduction about the respected speaker, Dr. J. Jayaganthan, Professor and Head, Department of Bioinformatics, Alagappa University, Karaikudi.
respected dignitaries, the scientific communities who have joined online, faculty members, and my dear student friends. Good afternoon to one and all gathered here. As Mahatma Gandhi said, education is the most powerful weapon which you can use to change the world. With his powerful quotes, it gives me immense pleasure to introduce the guest speaker for the plenary lecture 3, Dr. J. Jayakanthan, Professor and Head, Department of Bioinformatics, Allahappa University, Karaikudi. He is a man of excellence and perfection. He has 11 years of teaching and 28 years of research experience. He has qualified his PG degree in physics and did his PhD in crystallography and biophysics from University of Madras. He served as a CSIR SRF in the University of Madras, Chennai. He did his postdoctorate in Indian Institute of Science, Bangalore, and started his research career as a research scientist in Riken Harima Institute, Japan. And from the year 2010 until date, he is a professor and head, Department of Bioinformatics, Allahappa University. He is guiding 4 PDF scholars, 11 PhDs, and guided 8 MPhil scholars. His responsibilities are, he is the Director, Center for Internal Quality Assurance Committee, Directorate of Online Programs, Coordinator, DST PERS, DST FIST, Member of Syndicate, Chairperson, School of Biological Sciences, Coordinator, UGC Innovative Program, Member of Senate, Chairman, Board of Studies, Bioinformatics. His area of research is in structural biology and biocomputing, an area of specialization in small molecule and macromolecule X-ray crystallography. His current research focuses on structural and functional studies on vital drug targets, development of tools and databases in web-based search engines for analyzing macromolecular interactions. His distinctive achievements and award is awarded Tamil Nadu State Council Award by Tamil Nadu State Council for Science and Technology 2020, UGC Research Award, Fellow of Academy of Science, Chennai, Postdoctorate Fellowship, DST, DBT, and IRPHA, IUCR Young Scientist Award, Young Scientist Travel Award by DST and UNESCO, Research Fellow Award by CSIR. His academic body in other universities and institutes are Chairman Board of Studies in Bioinformatics, Bharadas University, Trivallur University, Bardiyar University, Coimbatore, Member and Board of Studies in Faculty, Bio and Chemical Engineering, Satyabama University, Chennai, Sastra Deemed University. He has delivered 226 special lectures and evaluated 37 PhD theses in his career. He has published more than 100 papers in international journal with high impact factors with 1,800 citations. He has attended more than 226 conferences, workshops, seminars in his career. We thank you, sir, for kindly being our guest, and we fully appreciate you finding the time to be with us despite your tight schedule. Our guest speaker is truly a renowned signer. Sir, it's a great honor to have you as a guest speaker for the International Virtual Conference on Biological Innovation and Computational Exploration for Pandemic Challenges, BICPAC' 2022. It's my indeed privilege to welcome Professor Jayakanthan to deliver the lecture. Hope a knowledge multitude increase with your talk. Thank you very much for the opportunity. Thank you.
Thank you very much for your nice introduction. It is my great pleasure to be part of this program. I thanks to the organizer and the principal and other college authorities. So this is my title of talk is the structural and functional studies on transcriptional regulatory proteins from Pyrococcus paracousi using in silico and in vitro approaches. I hope that it's not moving. Yes. So I hope maybe you are all well known about the structural genomic project. It was started in 2000 time. So all the countries working on different organism finally to match with the human genome project. So based on that, our India also working in Mycobacterial tuberculosis and also other organism to find out the to solve the crystal structure and find the function of the protein and propose the mechanism of the protein. So based on that, so in our lab, we are concentrating various organisms. In that, today my talk is giving on the Pyrococcus corycosi, which is the member of Hyperthermo plague, which is isolated from Japan in 1992. It is a highly stable organism from 90° to 105°, and which is the highest among all members of RK, for which the entire genomic sequence has been determined. So here, many kinds of amino acids have attracted and attention as biological regulators for the organism, which are used in their survival. So based on that, this protein we have selected from the Pyrococcus corycosi organism. There is a protein like PH0140 protein, which is initially uncharacterized protein. So we don't know about the function of the protein. So then we have selected this protein, that is a genome size is this much of genome size, and also which is coding the 2600, and it contains the more than 90 amino acid metabolism proteins. So based on that, this protein is uncharacterized. So we taken for our structural studies. So initially we have attempted in silico approaches. So these are my objectives. Go for in my talk. Like the first objective is transcriptional regulatory mechanism of tryptophan protein using extensive in silico analysis. And once we have attempted in the in silico approaches, then we have taken this same protein for our structural studies using the X-ray crystallography. Then also we have identified some DNA which is needed for this protein in the transcription. So we have used this one. Just a minute. எதுவுமே வரலையா? அப்ப நான் இப்போ போட்டேன் அப்ஜெக்டிவ்ஸ்ல இருக்கேன் நான். Presentation you பண்றேன். Now you can see now. Yeah. ஆமா இது எதுல மோர்ல இருக்கா? நான் திரும்ப க்ளோஸ் பண்ணிட்டு வரேன். Option Yeah. This is my title of talk. Can you able to hear? Audible? Audible? Hello? It's a structural functional studies on transcription regulatory protein from Pyrococcus corycosi OT3 in silica and in vitro approaches. Whether can able to audible? கேக்குதா மேடம்? Okay, okay, fine. Yeah, this Pyrococcus corycosi is a member of the hyperthermophilic, which is isolated from Japan in 1992. And this strain is hydrophobic, and tryptophan is recovered for the growth of this organism, and which is a highly stable from 90 degree to 105 degree. That's why we have selected for this organism to go for the structural studies. Now, it is a thermal stability of the genome product, and also it has the many industrial application of this protein. Actually, we have selected this protein from the Pyrococcus corycosi organism, which is a protein like coding the PH014 protein, which is an uncharacterized protein. So, which is not solved the crystal structure. That's why we have taken this protein for the our macromolecule structure determination. But during the parallelly, what we have done, so we want to confirm everything. So we have went for the in silica studies. So these are my objectives: the transcriptional regulatory mechanism of tryptophan protein using the in silica analysis, and also after that, so we have taken for this protein for cloning expression and purification and the structure determination of and the biophysical character of this protein. And then we have done for this DNA is very important needed for this protein. We have used the DNA through the in silica studies. So first I will go for the first objectives. This is the materials method. So we have used this protein for modeling and active site prediction, and also dynamics, and also to find the which is the suitable amino acid for the transcription regulator, and also we have done the all the in silica work. So this is a model. So we have taken this protein, and which is sharing the very less than 30% sequence similarity we compare with the other reported structures. So we have taken this protein and we constructed the model using the homology modeling. And once we have done this one, which shows the two domains, one is the DNA binding domain, another one is the effector binding domain. Also, we confirm with the using the Ramachandran plot. So all the residues are in the allowed regions and disallowed regions. So here it is showing there are 6 helices in the both the domains. So based on that, so we have taken this one, and then we needed to identify the our active site packet of this protein. Then and also we have done the before that. So we have taken all the reported structures using the multiple sequence alignments, and we have identified the similarity of the proteins from the other organism, which is showing the like you can see here the structures based on sequence alignment performed using the ESP print. The active site residues are indicated in the red colors. So based on the reporter structures, and also there is a DNA binding domain which is shown in the red color box. and a effector binding domain, which is showing the blue color box. So based on that, we have further taken this analysis, and we went for the finding the active site packet. So before that, we have done the molecular dynamic simulation. So here you can see here in the pre-simulation, so it is showing that both the domain is showing the 28 Armstrong. So it is a very huge distance. So and then after the 100 nanosecond, we have taken this protein, and which is closely showing that 8 Armstrong. That means so the structural transition of the two domains converged from the 28 to the 8 Armstrong. Here you can see the structure is very stable after the 20 nanosecond onwards. Then, so once we have done everything, so after the molecular dynamic simulation, we compare with the reporter structure which are available from the other organisms. Here we have taken the four protein structures from various amino acid binding the structure, and taken the like glutamine complexes and the methionine complexes and the lysine complexes. We have superimposed with the reporter structures with our structure, whichever we model the structure. So based on that, we have confirmed that there is a 4 active site packet of this protein. You can see here there is active site packet 1 and active site packet 2, active site packet 3, and active site packet 4. So these 4 active site packets of this protein are highly similar with the experimentally reported structures, which is represented by the, you can see here the dot view like that. So based on this active site packet, so we because here for this protein is needed the amino acids. The exogenous amino acids very important for the function of the protein. So based on that, we have identified the active site. Then we have taken the 20 amino acid. We have taken the 20 amino acid. We have went for the docking for the each active site packet. Here you can see here each active site packet we docked with the 20 amino acids, and active site packet 1 and active site packet 2 and active site packet 3 and active site packet 4. So based on that, here this protein is, you can see here, based on docking score and docking score, energy and everything. So the tryptophan is the needed for this amino acid for the function of this protein. So we have taken the tryptophan for our further studies. So then here one more important here, which is a DNA. DNA is needed for this protein. Then we have taken the DNA from the reporter crystal structure. So then we have docked with the DNA using the HERDOC server. So here you can see how our protein with interacting with the DNA. After that, so once we identify what are the residues are involving, here you can see this side. So interacting residues between our protein with the DNA. Based on that, then we have taken this result. So here you can see here how it is interacted with the DNA with our protein. Then we have taken this. Here you can see here. Then we have taken, so whichever we have identified from the in silica, there is a tryptophan important for this protein. Then we docked with along with the tryptophan and the DNA. Here you can see the interaction of the tryptophan with the protein amino acid. Here you can see the major group and how it is interacting with the active site packet 1 and 2 and 3 and 4. So based on this, the 4 amino acid, 4 tryptophan amino acid binding the active site packet. So we have mentioned that that this active site packet 1 and 2 and 3, which is the tryptophan is binding into the protein. So after that, then we want to confirm whether any other things happen. Then we went for the mutation studies like alanine studies. So we have mutated for the aligning the active site packet like active site 1 and 2 and 3, 4. Here you can see the tryptophan is showing the value. So based on this, then we have taken all this one and we have done for the all superimposed all the amino acid. So with the DNA and the protein, here you can see the binding of the exogenous amino acid in 4 different active site packet of the tryptophan protein in this along with the DNA and the active site packet of all the exogenous amino acid. So next, what we have done, so then we have taken all these, so we have done the molecular simulation because we need to check the structural stability of the protein. So here you can see the native, first we have done the native DNA 100 nanosecond, then after that we have docked with the DNA with the protein, DNA with the protein. Then whatever we have identified the tryptophan amino acid in all the active site packet, you can see the first active site packet protein with the DNA. So here you can see the RMS deviation is reasonably very stable. And also we have checked with this residues also. Here you can see the residues fluctuation. So based on that, so our structure is very stable along with the complexes of the DNA and the amino acid protein. So here, after that, so we analyzed everything. So these are the active site packet of the DNA and the tryptophan residues interactions. So here you can see the table. These are the interaction residues of the tryptophan DNA interaction, and these are the exogenous amino acids of the tryptophan along with the protein. So based this. And also from here you can also see the DNA is interacting with the which amino acid like serine 32, like serine in the active site packet 4 also is a serine 32. So which are found to be similar with the reporter structure also. So based on the reporter structure, we predicted which is involving is a phosphorylation mechanism. So then after that, so we have observed the closely we observed there is a arginine 28 residue is very important for binding and recognition of the DNA. Here you can see here how this protein and DNA interacting to each other and the role of the arginine 28. There are 3 helices, the helix 1 and helix 2 and helix 3. The arginine is once interacted, the arginine residues interacted with the DNA, that will open once open this groove is opens when the arginine 28 interact with the DNA. So if the arginine 28 is not interacted with, then it is like a closed conformation. So there are these observations we find out from the in silico studies. Here you can see how the open state to how it is converted into the closed state of the along with the DNA with protein. So after that, so we have taken for confirm with our DFT analysis. Here you can see here our the homo-lumo values, which is like nucleophilic and electrophilic attaches in the charge transfer reaction of the homo and lumo are the vital front layer of molecular orbital, which is play a major role in the chemical reactivity and also the stability of the molecules. So based on that, we also confirmed this how this electrons donation from donor to acceptor. So here you can see this DFT analysis. From this, we have confirmed the tryptophan as well as the based on the homo-lumo studies. Then here you can see this overall how it is working in the mechanism. Here you can see the DNA binding protein, the biological nature, and the effector binding domain. You can see here how it is making the open and close conformation based on the interaction of the residues, original residues. So once this amino acids and whatever we identify that the amino acid, tryptophan amino acid from the our in silico analysis, the amino acid, the tryptophan amino acids and the DNA molecule then bind with how the mechanism is happening. The DNA protein complexes, once it is bind with the protein and the tryptophan amino acid and DNA. You can see here the DNA protein complexes dissociation. And also in the active site packet 1. In active site packet 2, we can see here how this one is disruption happening in the transcription aspect. And active site 3, how it is transcription regulation is happening. And here you can see the fourth active site packet. So this is the observation we identify from our in silico studies. Then based on that in silico studies, we want to confirm this confirmation from the experimental aspect. Then we have taken this protein for making all the biological work like cloning, expression, and purification and the structure determination of this protein. Then these are the materials method. We have constructed the plasmid construction. Then we have transformed this in the PET-30A into this one and overproduction of the protein, because we need the large volume of protein to make a crystal to solve the structure determination. Using this is a method. Once we got the confirm our protein, then we went for all the like making the crystals using the various method. Once we got the crystal, from the crystal we need to go for the to collect the data using the X-ray diffraction. Once we collected data, then we can use all the kind of program to solve this structure. So what we have done the in silico work, we have taken the sequencing. From the sequencing, we have constructed the model. Based on the model, we identify the active site packet based on the reported structures as well as the sequence similarity of the reported proteins. So here the same thing, whatever we have done in the in silico studies, computational studies, the same thing we have taken this protein for the experimental studies. So these are the materials methods we have used. So here you can see this is we have taken the cloning vector PET-30A vector and the expression vector also, and this is our nucleotide sequence. The molecular weight of this protein is around closely the 18 kilodalton protein. So based on this information, we went for the So we have done everything, and we transformation and the protein purification of this protein, and which is a small scale. You can see here how our protein is conformed with the small scale, and it is showing that closely the 18 kilodalton protein. Then we have taken this one. Then we went for the gel filtration chromatography using this method. Then we confirmed our protein is expressing in the exactly our expected molecular weight. So then after that, so we have taken the protein. Then we have used crystallization methods. Various crystallization methods are there. So we have struggled a lot, and we have spent a lot of money to get the crystals for this protein. And there are many crystal screening conditions are also is there, and there are many crystallization methods are also is there. So based on that, so we used all the methods and crystallization screening, and finally we got the crystal using the hanging drop method. So using the hanging drop vapor diffusion method, these are the conditions. So from this condition, we got this very tiny crystal. Then we have optimized that crystals. We got the diffraction quality of the crystal. So then you can see here the crystal how we have got the diffraction quality of the crystal. This is the buffer condition. Which the buffer is needed for grow the crystal. So once we got crystal, then we have taken this crystal to collect the X-ray data. So we have taken this crystal, then we collected the data using the X-ray diffractometer. Here you can see the ABC value like alpha, beta, gamma, that is the cell parameter values, and the alpha, beta, gamma. So once we collected this data, so then we have used some methods to solve this protein. So we have used the molecular replacement method, because this protein is sharing the more less than 40% in identity. Based on that, so we have taken this one and we went for the solving the crystal structure of this protein using the molecular replacement method. So using the molecular replacement method, these are the data. So what are the things we have used for the the data collection like wavelength and temperature, what is the space group, and these are information we can get from the X-ray data during the X-ray data collection. Here is very, very important. Here you can see here the box. There is a R work and R free. That is the very, very important. So that means what we are doing in the in silica, there is a structural stability, the same thing. Here, once the R and R work is less the value, then your structure is very stable and your structure is solved. And also, I need to confirm the same way also, we have to run the pro check, find the Ramachandran plot and to confirm whether all the residues are in the allowed regions and everything. This is the structure we have solved from the experimentally. So once we got the structure, is automatically the structure incorporated the isolation in the protein, incorporated the isolation. So which is then we used the Ramachandran plot to confirm this structure. So like once we have done this one, then we have taken for to analysis of the structure. So as I mentioned before, there is a isolation amino acid binding into the active site packet. So which is come from the naturally incorporated in this protein, because this protein is needed the any kind of amino acid to make the function of the transcription function of this. So based on that, so from our experimental analysis, we observed, you can see here the electron density of the isolation residue, isolation amino acid. So you can see very clearly. So we observed, because this data is collected the around 2 Armstrong data, which is a high resolution data. And also we find the experimentally there is a 2 domain like DNA binding domain and the effector binding domain. So here you can see this isolation residue. After that, so here also you can see the interaction, how the isolation residues interacting with the protein. So like glycine 108 and the other interaction also. So we have observed. So based on this study, we have next we have taken with our experimental structure. So as I mentioned before, so already there is some reported structures are available from the various organisms which are the same related protein, but the sequence similarity is less compared to the like that is sharing the sequence similarity less than 35%. Some proteins are like 40%. Then we have taken all the structure, reported structure, we superimposed everything, because we want to see what are the difference observed from our reported structure as well as the in silica modeled structure and what we have solved structure. So here we identify there is the 3 observation. One like beta sheet deviation is there. Here you can see the one, number 1 is mentioned, there is a deviation. So once superimposed with our reported structure as well as the in silica studies modeled structures, you can see here the one is a beta sheet deviation is there. That means the RMS deviation is more compared to more than 1 angstrom. And the next observation, there is a observation in the C-terminal loop. Here you can see the second one, there is a C-terminal loop present adjacent to the effector binding. In compared to the other structure, there is no that kind loop not present in the reported structure. Then also we observe there is a third one, this additional helix present in our reported structure. So here also we have observed in the additional helix in compared to the reported structure. So then we have taken on this observation. Then we have taken this observation. Then we want to confirm everything using our computational DNA, because we have tried all the possibilities for making the complex with the DNA as well as the tetrapone amino acid, but we couldn't succeed the complex of this DNA as well as the amino acid. Again, we have taken the experimental structure. We have taken experimental structure. We used this methodology to go for the in silica studies, because we failure to solve the DNA binding along with the tetrapone amino acid. So once we have solved the native protein, but we cannot succeed in the DNA binding protein. So then what we planned, and then we have taken the same thing. We have reported success from we have taken the DNA and we used this methodology to making the complex with the our solved structure. So here we have identified there are many proteins available. These are the promoter region identification. We identified these promoters from the various proteins. Based on this promoter region, we in our protein we have used our protein for in silico studies. So we have constructed our DNA. Then we have taken this our protein along with the DNA, whatever we identify using the HEDOC server for protein DNA document studies, and we observed so what are the residues are playing this mutation, and we have also done the mutation for the instead of this one, we have done the mutation also to confirm everything. And then we have taken this structure for the like molecular dynamics simulation, and it is almost everything is you can see here the complex of the wild type and the mutation. This structure is very stable, and how this DNA is interacting with our experimental structure. So based on that, so we went for the analysis for the how this protein and the isolation, whatever is bind into the isolation in the active site packet, as well as the tryptophan residues. Here you can see the interaction between the residues and also the docking score of the isolation residues, as well as that tryptophan amino acid. So based on that, so we confirm this is essential for the isolation as well as any exogenous amino acid like that. We initially we thought, but before after that all the analysis, it is essential for the tryptophan as well as the isolation complex for this protein transcription regulations. So then what we have done in our protein solution studies, it is showed the our protein like is a monomer by octomer in the solution studies, which is showed the solution studies, but in the crystal studies, it is showing the monomer in the asymmetric unit. So based on that, so what we have done, we have generated the monomer is an octomer and all the subunits. So from the A to H, so we have made the octomer of the protein. Then we also designed the DNA molecule. Then we have complex with this DNA, our protein. So you can see here how it is wrapping the DNA wrapping and the octomer protein docking studies. So based on that, so here you can see the how much the distance between this like DNA 87.9 Armstrong. So once we have done this one, we have taken for the dynamic studies. We have done all the dynamic studies and everything. Here you can see how this isolation binding of the DNA along with our protein, and here also DNA along with the tryptophan. You can see here how this molecules binding with this tryptophan as well as the isolation, then we want to confirm the stabilization of this protein. Then we went for the molecular dynamic simulation with along with the DNA, protein, tryptophan, and also DNA, protein with the isolation residues. Here you can see we have observed here. You can see here the like conformation changes during the dynamics. The isolation, once isolation is bind, it is coming like that open conformation changes during the dynamics. The same way we also observed from the tryptophan amino acid. It is coming like the open conformation because this based on this, so then we come to the conclusion which is necessary for the like this tryptophan as well as the isolation amino acid. So after that, so we have taken for closely observation for other studies. Here you can see here we have done all the RMSs and RMS fluctuation and everything. It is like looks like the everything is a stable aspect. There is no any kind of fluctuation. But here you can see slightly there is a fluctuation in the active site packet of the particular molecule in the. After that, so we have taken here. You can see clearly. You can we come to the conclusion. Here you can see the DNA along with our octomer and how it is binding with. Then we can see the closed conformation to open conformation state, how it is changing. You can see here in the picture itself. Once you bind everything, then it is coming like open, like closed conformation to the open conformation state. Once this isolation bind, the same way also we observed that once the tryptophan complex bind, the closed conformation to the open conformation. These are the observations we observed from the computational studies, because actually we want to make it these studies in experimental aspect, that all the studies are going on, but now recently we have succeeded in the complex of this tryptophan and tryptophan. So we are also trying the DNA along with our protein. So far we didn't get it the good crystal, because we are getting the very very tiny crystals. So we are not succeeded to solve the along with the DNA. So maybe in the future, maybe we can succeed. So like this confirmation we have observed. Then, so here you can see here, so how this the negative charge surface of this closed conformation state, as well as the open conformation state of the octamer of this. So based on that, here you can see here how this charges, you can see the open and close conformation status. So after that, so we have taken here, you can see here how once the tryptophan once induced, it is coming like open conformation. The isolation also is coming like the open conformation. So these are observation we find from our in silico studies. Here you can see here how the DNA is interacting with that particular places. So based on that, then we went for this is the mechanism we have proposed in our studies using our in silico studies as well as the experimental studies. As I mentioned that, so far we have solved only crystal structure of this protein, but we didn't succeed in the DNA complex protein. So here you can see here this first one is our protein, which is from this study. Then once we have started the dynamics, it is closely interacting with each domain. Then once it is a DNA is bind into the protein, the based on there's the original residues, it is showing the open state and the closed state based on the interaction. So it is showing that it's open and closed state. And also we have studied the network analysis because of very less time. So I didn't discuss about the network analysis studies. Then we can see here, then we have based on our solution studies, it is showing that this protein is biologically, it is a octamer. Then we constructed the octamer of our protein. Then we, based on that, we designed the DNA. Then we made the complex with the DNA along with our protein. Based on that, so we also come to the conclusion. So it is like showing that it's open and close conformation of this our protein. So here we can see how it is the transcription regulation and oligomeric association happening in the mechanism of this protein. So from these studies, so we have identified, like you can see here, these are the in silico. So we have done the in silico studies using the homology modeling. Then after that, so we need to identify the suitable amino acid. So based on the amino acids, we have taken the 20 amino acids and we identify the 4 active site packet, like active site packet 1 and 2 and 3, 4, and we have done the docking with all the amino acids, and based on our docking score and energy, so we come to the conclusion that tryptophan is very important for this protein. So then based on that, so once we have done this one, then we have taken this protein. So we have collected the data in the 2 Armstrong resolution, and the structure is solved using the molecular replacement method. And there it is present in the isolation, is incorporated in the active site packet. Then based on that, so we observed our experimental studies. There are 3 observations. One is like a beta sheet observation, and another one is loop regions, and another one is additionally helix we found so compared to the reported success. So after then, so also we have taken the we are not able to succeed, and we go use the DNA for a complex DNA. So finally, we come to both the complex have the undergone the exogenous amino acid induced the open acromatic assembly and also interact with the promoter regions of DNA molecule during the dynamics. So presently, these studies are going with our complex studies with protein, and maybe soon maybe we will get the complex structure of the protein. Then only we can say about the mechanism of this protein. So these are the funding agency. So I acknowledge this funding agency for supporting my project. And here, these are the published. If you want more details, maybe you can check this two publication, which is published recently. Maybe you can take this publication. You can go through along it. Any kind of information needed, kindly to email, and also we can discuss through the or you can visit our lab also. So, and these are my students. This is a person, Dr. M. Richard. Is recently received the PhD degree, doctorate degree, and he is the persons working this protein. So, all in silico and experimental studies. These are my PhD students, and currently they are doing various protein working in this. And so I am really very happy to be part of this conference, and thank you very much. If any questions, please let me know. Thank you very much.
The session is open for queries. Thank you, sir, for your valuable speech. The lecture was really informative.
Thank you very much. Any question, please?
Is there any questions, dear students? Is there any, those who are connecting in online are requested to ask the questions. Sir is leaving for a meeting now. So I request those who are listening in online, you are requested to give your questions. Okay, thank you. So it's a time to thank and honor our keynote speaker, Dr. J. Jayakantan sir, who is serving as a professor and head department of Bioinformatics, Alagappa University, Karaikudi. Alagappa University is a university which is established by the government of Tamil Nadu, State Government Institute. Sir, thank you very much for spending with us for your valuable time. And also on behalf of the organizing committee, Department of Biotechnology and Bioinformatics, BICPAC' College, we humbly thank you for your presence through online. Thank you very much, sir.
Thank you very much.
Thank you, sir. Thank you.
Thank you, sir. Next part of plenary session starts by 4:15 p.m. I request all the participants to join the meet sharply at 4 p.m.
It's okay. Okay. I have joined it. Start.
Dinesh Raja, Assistant Professor, to give a brief introduction about the most awaited, renowned author and respected speaker, Dr. R.C. Dubey, Professor, Dean, IQAC, and renowned book author, Gurukula Kangri Vishwavidyalaya, University of Haridwar, Uttarakhand.
Respected delegates from all over the India, respected students, respected members of the faculty, respected deans of different divisions, on behalf of the organizing team, BICPAC'22, we cordially invite you to this evening session, first day evening session. On behalf of the organizing committee, we extend a very warm welcome to the famous, renowned author, Professor R.C. Dubey. We extend a very warm welcome to you, sir. I'm very much delighted and we're very much thankful for your presence over here to deliver a scientific talk with the delegates and our students. Dr. R.C. Dubey, he is serving as a professor and head, Department of Botany and Microbiology, Dean and Research, GKV, Haridwar. And he is serving as a chairman of Institutional Animal Ethical Committee, former Dean of Faculty of Life Sciences, Gurukul Kangi Vidyavadaya, Haridwar, Uttarakhand. And Dr. Dubey did his MSc and PhD in botany from the famous Banaras Hindu University. His specialization is microbiology, microbial biotechnology, and plant biotechnology. Dr. R.C. Dubey has got additional qualification, diploma in yoga from BHU, and also he has got a diploma in Madhyama in Srimad Bhagavad Gita. Dr. R.C. Dubey, a famous renowned book author, professor, Department of Botany, served as a professor in the Department of Botany with effective from 12th September 2004. He has served as a reader in the Department of Botany and Microbiology, Gurukul Kangi Vishwavidyalaya, Haridwar from 12 September 1996 to 11 September 2004. Earlier, he served as a lecturer in Common University, Nainital, Uttarakhand from 8 September 1987 to 1996. He has got a best research experience, 33 years of research experience, almost 3 decades. And also, Professor R.C. Dubey has got 33 years of teaching experience. He has produced 25 PhD. He has published 14 books with ISBN number and 173 research publications. At present, he is guiding 4 PhD students and he has received a certificate of merit from the Vice Chancellor of Banaras Hindu University. And he is serving as the life member and fellow of the Indian Botanical Society. He has attended many workshops and many conferences. He has completed more than 3 crores of major and minor research projects from all over the India and reputed research institutions and reputed government agencies. Moreover that, the students of biotechnology and bioinformatics all over the India, when you say you are using which book for your textbook, all the students will say that we are using the textbook of biotechnology by R.C. Dubey. So it's a privilege, it's an honor, and it's a great opportunity to interact with Dr. R.C. Dubey, a renowned professor, a scientist, and a renowned book author. Once again, on behalf of BICPAC'22, we extend a very, very warm welcome to our R.C. Dubey, sir. Over to Dr. R.C. Dubey. Thank you very much.
Thank you, Dr. Dinesh Raja, for nice words and introducing me to the audience. I am sharing my screen to all the audience to deliver a lecture on the topic which you have suggested me. Is it visible to all?
Yes, sir.
Challenges of living with COVID-19 and subsequent variants. This is the topic which I selected to give the brief idea. Though since morning you have been hearing COVID, coronavirus, so many names. So what is my opinion, my experience? I was informed to introduce something about the coronaviruses and subsequent effect. You know that the viruses are very old as far as their origin is concerned, because during the course of evolution, it was the RNA that got evolved first, followed by DNA, and obviously RNA viruses evolved first. Coronavirus is also RNA containing virus, so its origin is before the origin of any of the prokaryotic microorganisms. But you can put the question, earlier it was not known, where from all of a sudden it emerged and influenced the whole world. Globally, millions of the people lost their life, including India as well as Tamil Nadu. So this new strain of the coronavirus, it has become very much deadly pathogenic. Though the SARS severely acute respiratory syndrome, it was known since 1960, but several other waves came, and those waves were limited to only certain countries. It is the first report I know as far as I am going to the past, before the start of this 21st century, the SARS disease also appeared, but not more much were done because of the lack of facility and unavailability of tools and techniques of bioinformatics. So bioinformatic tools and techniques that developed during the last 2-3 decades, it helped a lot in understanding the full structural topography of the coronavirus. So in 2003 and 2013, the SARS-CoV and Middle East Respiratory Syndrome, means MERS-CoV, caused many deaths in different areas in different countries. But SARS-CoV-2, also called as COVID-19, which I pronunciate as China origin virus disease, COVID-19, it is spread in late December 2019 from wet market, where Laboratory of Virology of Wuhan Institute of Virology, China, is there. So this is the first report, and from here it spreads in many countries. In the beginning of 2020, it is spread from Wuhan Institute of Virology. China neither declared the total death nor informed the World Health Organization about the secret of this disease. In 1920, World Health Organization declared this disease as a pandemic disease. Now, the unique and novel genetic makeup of SARS-CoV-2 has created hurdles in biological research, due to which the potential drug vaccine candidate need to be discovered. My dear students, my dear friends, do you know? All of you know that this is RNA containing virus, and all the RNA containing viruses are very dangerous because of the presence of RNA as genetic material. You know that RNA very frequently undergo mutation, either through genetic shift or genetic drift, and due to mutation, the locations of the amino acid in the polypeptide get changed, and because of the changes in the amino acids in the polypeptide, there is shifting and nature of the the proteins, and that is why time to time, it takes the very dangerous shape and dangerous pathogenicity. The advantage of bioinformatics in viral research had created a milestone in the last few decades. Suppose if there would have been no bioinformatics and metagenomic approaches, it would have been not possible to produce the vaccine in a very short time, though the researches are going on. The bioinformatic tools and technique, as I told you, it involves next generation sequencing, genome-wide association studies, computer-aided drug design, etc. And these have effectively applied in COVID-19 research methodologies and discovered novel information on SARS-CoV-2. It has been found that more than 10,000 times a mutation have occurred in the coronavirus. Implementation of bioinformatic studies in COVID-19 research has not only sequenced the SARS-CoV-2 genome, but also properly analyzed the sequencing error, evolutionary relationship, genetic variations, putative drug candidates against SARS-CoV viral gene within a very short period. And these are the approaches where from nasal or oral inocula, by using the swab is collected and RNA is extracted, it is passed to RT-PCR, and then positive and negative, in the bands that it developed. Then RNA quality control, RSCQC, is analyzed, the cDNA synthesis is done, and these are several steps. Then ultimately gene sequence is done, and genome assembly variants, calling junction reads, everything is analyzed. And the scientists declare that there is a variation in the genome organization. So interconnected bioinformatic applications implemented in COVID-19 research, where, as I discussed, information gathered from different sources are being analyzed and reported. As far as taxonomies of coronavirus is concerned, human coronavirus, I told you in 1916, it came into existence. Though existence was there, it was reported. This coronavirus has been divided, human coronaviruses, into two groups, two subfamilies. Basically, it is called as coronaviridae. So the species is coronaviridae, family is coronaviridae, and the classes has been created, subfamilies into two, coronaviruses and tonovirus. The coronaviruses are put into two subclasses, alpha coronaviruses and beta coronaviruses. The beta coronaviruses includes both SARS-CoV and MERS-CoV. Out of the SARS-CoV, SARS-CoV-1 and SARS-CoV-2, to which it is called as COVID-19, which is highly pathogenic and cause severe disease infections throughout the whole globe. Now, studying with computer-aided, the model has been developed, and the structure has been analyzed by using electron microscopy, and it has found that this is enveloped virus. It has its envelope, and having non-segmented, continuous, linear form of plus sense single-strand RNA as genome covered by a capsid. Its genome consists of 4 structural proteins, and these structural proteins are very important. In any of the viruses, structural proteins are important. Out of which the spike protein, you can see here, spike protein consisting of S1 and S2 protein, then nucleocapsid protein, certain amount of proteins are associated with the RNA known as nucleocapsid protein, then matrix protein, which is a part of the capsid, then envelope protein. The envelope is also a part of the host that surrounds the capsid, and single-stranded plus sense linear RNA is located. So this is genetic setup and structural topography of this virus. Now, one of the important thing that is found in this virus that here the S1, S1, S2, and many more variants are now have been discovered. They recognize and interact with the angiostatin converting enzyme, abbreviated as AC2, in the present in the lower respiratory tract of a human. And this is the entry, the mode of entry. By taking the help of it enters inside the respiratory tract of human being. Therefore, ACE2 acts as a host receptor or molecular receptor. Now, single stranded RNA of messenger RNA is 29.9 kilobase pair long. Its genetic material is large size, 29.9 kilobase pair long, including 13 open reading frames. Open reading frames, not one or two. 13 open reading frames are found, and it has been given there. Right from here to here, you can see open reading frame. So the coronavirus genome has the longest RNA virus genome known so far. Its RNA-dependent RNA polymerase is also known to have proofreading function. This is unique feature found in this virus. Now, in the variants, the variants arises due to mutation, due to that form different lineages, and also form different variants. So mutations occur through genetic shift or genetic drift. So mutations are where this refers to a single change in a virus genome. This is in our genetic code. This is known as mutation. Lineages are those it has a group of closely related viruses with a common ancestor. For example, SARS-CoV-2 has many lineages, all cause COVID-19. All the lineages of SARS-CoV-2 cause COVID-19. And the variants is a viral genome that may contain one or more mutations. Just you see here. The mutation may be one or more than one. In some cases, a group of variants with similar genetic change, such as lineage or group of lineage, may be designated by public health organization as a variant of concern. The two terms have been given by World Health Organization. Variants of concern, VOC, or a variant of interest, VOI, due to shared attributes and characteristics that may require public health action. So, on November 30, 2021, the US government SARS-CoV Intergency Group, SIG, classified a new variant known as Omicron. Omicron as a variant of concern because Omicron is highly contagious. The making contamination is spreading speed from one individual to another individual is the highest. It has been found highest in this group, Omicron group. In earlier groups also, the Delta virus, it was also highly contagious. But as far as mortality rate is concerned, in Omicron, mortality rate is less than that of the Delta virus. I do not want to say anything about Delta virus because all of you have the bad experience of Delta virus last year. What happened? There was no oxygen cylinder in the whole country. Anyhow, the government of India managed and tried to save the life of millions of the sufferers. SIG variants classification scheme is that here the SARS-CoV-2 intergency group, it has a screen. It has been divided into 4 different types. Variant being monitored here: alpha, beta, gamma, epsilon, eta, iota, kappa, delta, mu, zeta. All these names have been given, and all the variants have been put inside it. Now, second is variant of interest. No group has been put so far. Variant of concern, 2 important groups. The Delta, last year that is spread Omicron. In these group, in Delta and Omicron, there is a variation, a slight change in their genetic setup and consequently in their polypeptide. Then variant of high consequences. So these four groups have been created by ESIG, SARS-CoV Intergency Group, formed by United States government. Now, World Health Organization, as I told you, have put name in a Greek word, alpha, beta, gamma, epsilon, putting different variants in this case that was that appeared at different intervals, and these are the intervals, date of designation. Now, as far as genome explosion is concerned, because genome explosion is very important to know to all the person who has joined here. Since 2020, the scientists have explored about 6 million SARS-CoV-2 genome sequence on data sharing platform, but many of them in past 10 months, 6 million SARS-CoV genome sequences have been shared on data sharing platform. These deposits in Africa alarm on the Omicron variant because it is spread very fast rate. In January 20, SARS-CoV started spreading. Then in December 20, alpha and beta variant is spread, then gamma variant, then delta variant, Omicron. Very recently, Omicron is reached to the whole globe, starting with South Africa, Asia, North America, Europe, globe, and none of the countries untouched with these viruses. The vector of infection, South Africa confirmed daily COVID cases by variant based on sequence submitted to GISAID database. You can see here in the beginning, the mixed population of the viruses got spread, followed by beta variant, then alpha variant, in between delta variant in the beginning of the 2021 in India came late, but in other countries like Britain or America, it is spread earlier. Then delta crossed the limit. It caused maximum mortality, and followed by Omicron that spread this year. Now, World Health Organization label Delta virus because the World Health Organization has given characteristic feature of the variants of this group. It has increased transmissibility. Nearly all lineages designated as Delta are susceptible to emergency use authorization, monoclonal antibody treatment, and not susceptible to some monoclonal antibody treatments, reduction in neutralization by post-vaccination sera. And the Omicron, it has attributes like potential increased transmissibility, potential reduction in neutralization by some UAE monoclonal antibodies treatment, and potential reduction in neutralization by post-vaccination sera. Now, SARS-CoV-2 variant wave that came in different countries, like in United States, as I told you in the very beginning of 20, there was a mixed population of the SARS virus or coronavirus, and later on in the beginning, or you can say July to October, then in January, in April, and April 21, then in July of 21, this is of 20, different group spread. In the meantime, when alpha virus was on the top, gamma virus also spread in April 2021. Then followed by delta virus, maybe April late or May or June, delta virus also is spread in the United States. United Kingdom situation is somewhat different, but all the virus variants were also present there, and there several mutations occur, and finally Delta virus also caused a very havoc situation. As far as India is concerned, in our country, in 20 July, October, January, there was a mixture of the variants. Then in March, there was a spread of the virus, and this virus infection, it caused this virus also caused infection, coronavirus known as COVID, and late it was known as in 2021 Delta virus that caused maximum mortality in India. But as far as mortality rate compared with the population with other countries concerned, the mortality rate with respect to population was less in India. In South Africa, you can see here the Omicron started developing in late December, and from there it spread to many more countries. So this is the first wave. This is the first wave, you can say, and first wave, and second wave, and third wave. So Omicron is third wave, but no virologist can take the guarantee there would be no fourth wave, because this is a virus and mutations are going on in all the viruses continuously. As I was dealing with in the beginning of this lecture, I told you that there is a ACE2 angiostatin changing enzyme 2 found in the respiratory tract of the human being. It combines with the protein, the spike protein, and immediately it enters in the endosomes. From the endosome, it is transported in the cytoplasm. Then RNA is released plus RNA. Translation occurs in the meantime. The early protein and late proteins are synthesized due to proteolytic activity. Endosomes are disintegrated. Synthesis of RNA occurs plus strand of RNA synthesized. Transcription also done. Then there is a packaging of the virus, and ultimately it is released from the cell membrane of the host through budding system. Through budding, and it spreads large in number. Wherever it has to go, it travels there and goes there. As far as fatality is concerned, with respect to infection rate and fatality, males were infected about by 73%, females by 27%. Maximum infection and mortality were caused in the male, followed by female of the different age group. You can see here different age group. This is religion, because most frequently males used to come outside from their home and females remain inside the home, looking after the activity of their houses and kitchens. Age-wide distribution of COVID, as far as hospitalization of 1,040,000 number of patients is concerned, again the persons of 21 to 30 years age, they were infected maximum. They were hospitalized maximum 25.84%, followed by persons of the age group 30 to 40 years, and then 41 to 50 years. The old person did not come their house because they were thinking that their immunity is weak. So the hospitalization possibility was varying from 2 to 6%. 2 to 6%, you can say. Even the percent of about age group of about 60%, the hospitalization cases were only 12%. Now, how India is dealing with the COVID-19? This is the major question. Indian government tackled very easily, very gently through taking different major and minor steps. First case was reported in Kerala. Then 2 more cases were reported. In the meantime, World Health Organization coined the term coronavirus, COVID-19. Earlier, it was known as coronavirus. Then Government of India in late March, I think it was 24, announced that one must maintain the social distancing. Then in March again, late, Janta curfew was declared. And first phase of lockdown was declared on March 23rd. First phase of lockdown was declared of lockdown, and 2 days before Janta curfew was declared. Then second phase was declared in April. April, second phase of lockdown. Then travel, visa, every restriction was done. Then in May 2, third phase of lockdown was declared. Then May 7, fourth lockdown, and several labs establishment, 2 lakh samples test landmark. Then testing and tracing of the coronavirus infected patient was started, because at that time there was no vaccine, no treatment, no medicine, nothing was available. So government of India tackled this situation very wisely, just announcing to maintain physical contact, physical distancing, and remaining their respective houses, and not coming out from their houses, stopping the railway, the flights, the buses, the taxis, the students, teachers, no person will come their houses. So these, and during this course, most of the people suffer a lot. Most of the people suffered a loss. Several people, they lost their life. And about 1 lakh COVID cases were reported by May 2022 in 2020. And ultimately, up to July 10, 8 lakh positive corona cases were declared. 8 lakh coronavirus cases were traced out. It was recorded, and there was no vaccine. Just imagine, no vaccine, number of corona cases are increasing. So this is the way how government of India tackled, not to increase the coronavirus infection, but to minimize the rate of infection. Now in September recorded the highest number of cases, 2 lakh 26 lakh 21,418 cases by September. Just imagine, number of cases were increasing day by day. And now in 2022, 1st February 2022, and the number of Omicron cases, they were maximum, they were on the top. And still it is, the number of cases is going down, and hopefully by 31st March 2020, this will come to the end. But I don't know, nobody knows what would be after March 2022, whether any fourth wave will come or not. My dear student, my dear participant and friend, different symptoms of COVID variants were reported. Different symptoms by different variants were reported. Some common symptoms were fever in 80 to 99%, loss of appetite in 40 to 84% people, fatigueness, loss of smell 15 to 30% of individual, not all. Shortness of breath 31 to 40% of individual, cough by 59 to 82% individual, coughing up sputum 28 to 33%, muscle ache and pain 11 to 35%, difficulty in walking, confusion, blue face or lip, coughing up blood, persistent chest pain, decreased white blood cells, kidney failure, high failure, and lastly, death due to multi-organ failure, septic shock, and blood clot. We are also hearing daily that the people, some of the patients, those who were infected by coronavirus last year, they died this year. It means it has effect, post-infection effect, even after 1 year. Maybe that after 1 or 2 years, the symptoms may occur. The septic shock may occur. Nobody knows. But as per doctor's suggestion, they also say that anything is possible if person is having the corona infection. Now, you know that several step measures are there. If a person is infected by this virus, certainly he or she will sneeze, and through sneezing, the droplet nuclei of different size comes out, and another person receives those droplet nuclei where millions of the particle bearing corona particle are attached. So the droplet particle from one person to another person transmits through breath, through cough or contact. Now the size decide who will be infected. For example, and the style of sneezing also decide the distance to be traveled by the different droplet nuclei. If a person is there, for example, during the sneeze, if he or she sneezes, keeping their head straight, the droplet nuclei will spread vertically. And this is the size of droplet nuclei are very small, it will travel away up to 6 feet of distance. And if the droplet nuclei are larger in size, it fell down immediately, traveling to about 3 feet distance. So it is the size of the droplet nuclei and direction of the neck that decide the person standing in front of you shall be infected by the virus or not. Now, different other measures: washing hands, use of sanitizers, making distance, mode of sneezing, and maintaining distance. These were suggested, all of you know. And lockdown situations, now another way is shaking hand was also prohibited. And this is the culture of India. We always say vanakkam, folding both of the hand in front of you. We say vanakkam, namaste. It means that another infected person will not come to your contact. So this is the oldest tradition of our country. We say pranam. This is known as pranam ashan or Anjali mudra. Anjali mudra. Any of the Bharat Natyam dance starts with Anjali mudra. Bharat Natyam dance starts with Anjali mudra, where there is a folding of the hands. So this Anjali mudra or pranam ashan is the Indian culture. Namaste means I bow down to you. I bow down to Namaste. Bow down to you. You means the divinity that resides inside you. You does not mean a person. The divinity, the power, the divine power, the purity that is present inside you, I bow down before you, means the divinity. This is the meaning of the namaste. Now 6 feet the distance. If I told you, if you are having a longer distance, then it is not possible to come the particle of the corona. If distance is less, then whole of your body, right from head, even upper side of the head to your vest, will be covered by the coronavirus particle. So as far as social distancing is concerned, in the very beginning, all the persons residing in the city, they were following and making social distance. But soon after, there was a rush to pick up the train, to pick up the bus, to get stand in queue in the line. No social distancing. You can see, and still today, people say, where is your corona? I don't know corona. No corona is there. It is the propaganda of the government. My dear friend, this is statement given by those persons who are not infected or who are not scared by the coronavirus. Many more protections were given, recommended to the frontline workers. The use of PPE, mask, N95, goggles, hand wash, and use of any type of hand wash sanitizer having 75% ethanol. According to Dr. Soumya, Swaminathan, who is working in World Health Organization, she said that coronavirus, it has entered in society, it will not eliminated. COVID-19 cannot be eliminated even up to 5 years, even after vaccination. This is her statement. And it appears that several other type of waves may come in the future. Then what we have to do? We have to go for vaccination. Vaccines were developed last year. It was started from from February or March, and most of people have been vaccinated. But my dear friend, keep in mind that vaccines reside inside your body. Triggering of natural immunity is inside you. Medicine of coronavirus is within your body. Whosoever have been cured, when there was no vaccine, those persons who were cured only because of their own immunity present in your body. How is it possible to increase the immunity? There are certain precautions, the food materials. You have to perform daily yoga, exercise, or any game. Homemade pure vegetarian food you have to consume. Pure vegetarian food, fruits, especially sour and fruity juices like orange. Uh, it is recommended. Green vegetable like spinach, radish, chinopodium, it is recommended. Dal, good, jaggery, pure vegetarian oil, mustard oil, olive oil, linseed oil, ground nut oil, coconut oil, all these are recommended. Tulsi, Indian basil, and other medicinal drink, it is recommended. Pure milk-based product like lassi, chach, curd, milk, it is recommended, not any artificial. And performing daily yoga and exercise. What we should not take? We should not take the maida-based product like bread, naan, datura, bhatura, burger, pizza, samosa, kachori, pav. All these are not recommended. Refined oil is not recommended. Sugar and sugar-based product must be avoided. Any junk food must be avoided. Food cooking in aluminum vessels like cooker must be avoided. Cold drink and packed food must be avoided. These suggestions have been given by the experts. Now I want to give some of the important points that this coronavirus or any type of viruses, are they new diseases? No, such type of pandemic have also come in the past as well. Don't think that this is a new disease. Certain viral diseases have also come in the past. Just you look this baby, on his face there is a appearance of blisters, and these blisters are of the smallpox. There was a time when there was no cure of smallpox, no cure of mumps. I want to put a question to you. Just you remember, in Tamil Nadu, there are many Mariamma temples. Many temples with the name of Mariamma, and you people used to go there. This similar type of temples are also located in North India, known as Shitala Mata temple. Shitala Mata, Mariamma. This is the symbolic of the goddess Parvati. Mari Amma, Parvati, goddess Parvati, symbolic temples. So these temples are symbolic of smallpox. When smallpox spread during 1,000 years ago, there were no medicine. Only these temples and folklores saved the life of millions of the people, not in India, in Europe as well. In Europe, Mariamma temple is known as the goddess of smallpox. Goddess of smallpox. Again, there is a name, goddess. In China, smallpox is known as beautiful flower. So in different country, in Bangladesh, it is known as Ola Bibi, Bibi Ma. In different countries, there are different names. Similarly, the disease which is known as mumps, It is known as mumps in English, but in Sanskrit known as Galganda. Galganda Graha Ghora, E Chante Darunam Nidam. In Skanda Puran, there is a mention of these two diseases. Galganda Graha Ghora, here Ashtakam Shitala Debya, Visfotak Bhayam Ghoram, Grihe Trasya Jayate. In Skanda Puran, it has been mentioned where there is a smallpox or mumps, we should not go to the home of that person. It means we must maintain social distancing. And this can be cured by making social distancing, and this is a measure of a hygienic condition. You see, mumps is here, which is known as Galganda. In Atharveda, there is a mention of mumps, Galganda, with the name as Apachitam Lohini Nam, Krishnam Ati Sushrutam. In Atharveda, there is a mention, and mumps is called as rubella virus. Rubella virus that belongs to minor sense RNA containing virus of paramyxovirus group. It causes mumps in human beings. Now this disease has been cured only because of development of vaccine known as mumps, measles and rubella. MMR vaccine is available in Tamil Nadu as well, in whole globe, in all the country. This vaccine is available with the name as MMR, which is used for mumps, measles and rubella. Now, these germs and viruses can also be killed if you are sitting, you are taking this sun bath. Apply oil, mustard oil on your body. Sit in front of the sun rays for about 20 minutes early in the morning when the color of the sun rays are a little bit red. When there is a reddish color, then all the germs on your body will be killed, and such person will not suffer for any microbial diseases and the heart shock. It has been mentioned in Atharveda. Go rohitasya varnena tena tva paridadmasi. Now, there is also mention that we must change our habit. We should not take out salt, ghee, oil, food, water and milk without washing your hand. When your hand is properly washed, only you can take it out. Otherwise, you should have to take it out by using a spoon. Without using a spoon, you should not take it out. Salt, ghee, oil, food, water and milk. In Dharma Sindhu, it has been mentioned, Lavanam Benjanam Chaiva, Gritam Tailam Tathaivacha, Lehayam Payam Cha Vihitam, Hastdattam Na Bhakshayet. Manusmriti said that without any reason, you should not put your finger in your eyes, in your nose, in your ears without any reason. Anathura swani khanicha is nimittinan. Otherwise, the microbes attached on your hand will enter your eyes. And you should not take meals without washing your hand, feet and mouth. Hastapade mukhe chaiva panchadre bhojanam charet. You must take food material only after washing hand, feet and mouth. It has been mentioned in Shushruta Samhita and Padma Puran. Then you should not wear the cloth of others. Don't use the cloth like towel used by other person for drying your body after a bath. Na dharate paravastram snanavastram kadachana. In Sanskrit, it has been mentioned. And cloth once you have worn, it should not be worn again and again. It means after wearing your cloth, it should be washed properly. And if you are changing the cloth, it is not visible. It has not been suggested in our old and ancient texts. Some precautionary measures as far as wearing the cloth is concerned, you should not wear the cloth of others. Don't wear the wet cloth and don't wear others' cloth. Now, my dear friends, ladies and gentlemen, and students, all these are very interesting. That the virus disease is not known. This is It is very old one, and several viral diseases has occurred. It has caused pandemic in the past as well, and during last 2 years, all of you are suffering such viral diseases, and I don't know that in near future viral diseases will another wave of viral disease, coronavirus will come or not. But my advice is that whatever it has been mentioned, either by Government of India, frequently wash your hand after 20 minutes. But I used to say, if you are not washing after 20 minutes, at least you must wash before eating the food material. Don't put your fingers and hands, your mouth, in your eyes, in your nose, in your ears. All these suggestions have also been given in our ancient textbooks. So thank you so much. If you have any query, you can put your question. I had widely traveled in Tanjore, in Kanchipuram, in Bharati Dasani University, but I have never come to Trichinapalli because Dr. D. Raja did not invite me to deliver lecture in his college. But I assure, if there would be time, I will come. I have come to Madurai Kamraj University also, in Salem. I have many more friends in Tamil Nadu. So thank you so much. And if you have any query, you can put question to me. Now, over to organizer. Over to organizer.
Very good evening, sir. Thank you very much for your efficient way of interaction with our students. Surely our delegates and students have experienced the bright way of COVID and other active things. Sir, I want to ask one question that Dr. R.C. Dubey is well known by the way which you have authored a book called The Textbook of Biotechnology. Sir, can you tell me how you have got the idea to publish such a book, and how this book has been widely used all around the India for all the students are using your textbook? Can you tell me the secret behind this?
Yeah. I am telling you, if any challenge comes to you, don't go away. Don't run away against any challenge. When I joined Kumai University in Nainital as an assistant professor for the first time in 1986, my head of the department then told me that you have to teach biotechnology because you have come from Banaras Hindu University. And surprisingly, at that time, no teacher, there were 10 teachers. I was the junior most. No teacher came forward to help me. All the teachers left me to teach biotechnology. And my situation is that I was not taught biotechnology. I did not know ABCD of biotechnology because that time there was it was not put in the syllabus. I passed MSc in 1981. And in 1981, in none of the university started biotechnology teaching. None of the college. I was not knowing what is the definition of biotechnology. But when my senior and elder head of the department told me, you had to teach, I got scared. But I prepared. I told him that, sir, I will certainly teach the student the biotechnology. I will try my best. That time there was no internet, no mobile, no telephone, no facility. I am telling you the story of late 1980s. I went again to Banaras Hindu University and stayed for 5 days in the hostel and spent 10 hours per day in the hostel. For 5 days, it means 50 hours I spent in the library, and I collected a lot of material. There was no photo stat facility. I wrote everything on the paper, even the material, method, introduction, what is this, what is that, what is restriction enzyme, what is genome. I did not know. I collected lot of information for about 1 year and prepared note, started teaching to students, and my name is spread in whole of the university as well as associated degree colleges. In whole state, I become very famous without writing the book. I wrote the book after 2 years later in 1989. In 1989, no book on biotechnology was available. So I became very much famous because it is the only student that increases the name and fame of any of the teacher. Then I got stimuli, inspiration from my student that I am doing good. I contacted the publisher and very easily he gave his consent by sending inland letter, no telephone, no email. I received it and I submitted the manuscript, and by late in 1992, my book of biotechnology was published. So this created me interest that being as a teacher, I am going to contribute something. I felt very much proud that in spite of teaching my student biotechnology, I have contributed in the country to those poor students who are unable to understand the very complicated English book written by foreign authors. That is why you might be seeing, might be experiencing that in my book, I have used very simple language and made the whole book very illustrative, drawing the diagram by my own hands. Some of the diagrams, they are not so beautiful, but those diagrams gives inspiration to me. I used to say to my student, just you see, this diagram I have drawn by my own hand. So this is a story when I became very famous and became a book writer. A new book I have published very recently, Vedic Microbiology. The microbes that their names have been mentioned in Vedas, and extracting some of the information. Just now I explained those shlokas and mantras mentioned in Vedic microbiology. So those belongs to my book itself. So thank you so much for asking very curious question and sending me back to my early age of this teaching. Thank you so much.
Thank you very much. Thank you very much, sir, for your kind acceptance to be as a scientific speech. Thank you very much, sir. Thank you on behalf of the organizing committee. Once again, we thank you very much for your presence. Thank you.
Any query from the students? Any question from the student? You know, when I used to South India, in any of the institution, then student surrounds me and put a lot of questions. Ramya Mohan, do you have any question? All are hearing to me? Archana Sharvanan, Anjurani, any question?
Question, sir. She is Miss Neka, sir, students of second MSc Biotechnology. Okay. She is the president of our Helix Association.
Thank you for your informative session, sir. It gives us immense pleasure to be a part of BHC's conference, BICPAC'22. And my question is, what will be the next fourth and fifth wave of COVID spike protein characteristics if it exists?
Again, pardon me, I am unable to hear. Can you repeat again?
Sir, what would be the characteristics of the spike protein of COVID if the fourth and fifth wave exist of the next generation? Sir?
I cannot say exactly what will happen. It depends upon the It depends upon the aggressiveness of the virus and the position on which the changes have been brought about. This is the whole summation of the effect of the changes that has been brought in the viruses. No any single mutation or single change can cause such type of havoc. It is the whole holistic one.
So thank you, sir.
Thank you very much, sir, for your kind presence. Now is a time to listen the first international speech. You are going to have a very good experience. Now I request Dr. Bliss Ramya John, the Assistant Professor of the Department of Biotechnology and Bioinformatics, to introduce the international speaker of this two-day international conference on biological innovation and computational exploration for pandemic challenges. Please, ma'am.
It's my immense pleasure to welcome on behalf of Department of Biotechnology and Bioinformatics, organizing committee members of International Virtual Conference. I am very much proud and happy to welcome Professor Dr. Christina Alexandra Tringali. First of all, I thank you for accepting our invitation to this international virtual conference. She is an eminent researcher, a dynamic person with sincere inner efforts taken in her research career. She completed her high school graduation in the year 1992, and she completed her master's in the year 1998 in biological sciences, University of Milan. She had completed her exam for the license to practice as biologist in the University of Milan in the year 2002. In the year 2005, she completed her post-graduation in medical specialization in clinical biochemistry, University of Milan. Her work experience, she got her laboratory training as undergraduate student in the University of Milan. In the year 1998 to 1999, she had laboratory training as a postgraduate student at Milan University. From 1999 to 2006, she was a scientific technician at Milan University. From the year 2006 to 2016, she was working as assistant professor in clinical biochemistry and molecular biology, University of Milan. From 2016 at present, now she is currently working as associate professor in clinical biochemistry and molecular biology, Department of Medical Biotechnology and Translational Medicine, University of Milan. She has a very wide experience in various research teams all over the globe. Since 2006 to 2018, she was a member of the PhD in Biochemical Sciences, University of Milan. Since 2018, until now, she's a member of the PhD in Experimental Medicine at University of Milan. Our research activity focused our interest towards such studies concerning the control of cell glycosylation in health and disease. In particular, she deepened the role of enzymes involved in sphingolipid metabolism and sphingolipids in relation to cell signaling and creep processes, including differentiation, tumorigenesis, Crab leukodystrophy, and demyelinating disorders. She has a lot of publications in various international journals. I once again extend a warm welcome to our international speaker, Professor Dr. Christina Alexandra Tringali. Over to Dr. Christina.
Okay. I sincerely thank all of the organizing committee and you, Bliss, to invite me today and for this kind presentation. So I try to share my screen. Okay, just a moment, because Now, now I think you are able to see my screen. Do you confirm?
Yes, ma'am, we are able to see these.
Okay, thank you. Okay, here you can see just some famous views of Milan, the city where I live, and inside this is Lita Palace, where I'm performing my research and my work. Today I will present you our data about crab leukodystrophy. That is the topic of our current research. Crab disease is classified as a rare disease. In fact, about rare disease are so called because they involved very few people if we consider a single disorder. But if we consider the totality of rare disease, you can see here that about 400 million of people worldwide are involved in this type of pain. We must underline that the life with a rare disease, even in the best of times, is very difficult. Patients and caregivers report significant care inadequacy. Above all, the diagnosis could be very difficult, thinking that some diagnoses could belong for about 4 years, and in some cases that occurs before these patients have a diagnosis. Also, the type of treatment for this type of disorders are not known for all of them. Here you can see that above 70% of rare diseases are genetically inherited. Also, crab disease is among these. A small percentage of rare diseases can be caused by other factors like also infections. Consider that also SARS-CoV-2 is able to induce in children a syndrome known as Kawasaki syndrome that is classified as a rare disease. So also SARS-CoV-2 can be the source, the origin for other types of rare diseases. But see above all that we have a therapeutic opportunity only for the 5% of rare diseases. So many of these diseases are fatal just in childhood, or can be chronic and progressive. The impact of COVID-19 pandemic on all rare diseases was devastating, because for different reasons about at first, thinking that diagnoses were further delayed and proper treatments were not assured. Also, in some phases of the pandemic, drug availability was not assured. Maybe you remember that at the beginning of the pandemic, hydroxychloroquine was employed to treat COVID-19. Well, hydroxychloroquine is used for the treatment of some rare autoimmune disorders, and there was a problem in the availability of this drug. Another important thing is that also rare disease patients are being affected with SARS-CoV-2, and different complications related to their physical health and also to the possible treatments that they use can occur. Also, COVID-19 vaccination can be a challenge because it is recommended that these people are vaccinated. But we must underline that there are no clinical trial with the effect of COVID-19 vaccines in this type of patients. This is because we haven't enough rare disease patients for clinical trials. Adaptation to the new normal means that above all, telemedicine services could be employed in this period to support these patients. But we know that not all countries can give this type of service. And at last, our challenge that concerns research of rare diseases, consider that studying rare diseases is not easy. It's not always easy because there are many challenges related to funding, related to collect a cohort of patients. Also, to have biological samples. In Italy, we can enjoy the Teleton Biobank, and so we can today use samples like blood or also fibroblast cells isolated from these patients. But it could be very difficult to collect biological samples from this type of patients. And during this pandemic, troubles were higher because most of the research moves to COVID-19 and also grants and funding. So in this context, we are working also today on Krabbe disease that is called also globoid cell leukodystrophy. This is a disease that was discovered in 1916 by this researcher, Krabbe. This was a Danish researcher who identified the typical signs of this pathology in children. So here you can see that Krabbe disease can present in two forms. The early onset type and the later onset that involves later childhood or adolescence or also adulthood. So the early onset type of Krabbe disease is the most common, accounting for about the 19% of patients, and is also the most severe form of this disease. Babies who have early onset Krabbe disease typically develop future in the first 6 months of their life. And in this case, Krabbe disease is fatal, with an average survival of about 2 years. If the onset is later, maybe in later childhood, the survival can increase to 4 or 5 years. Here you can see the symptoms, the typical symptoms of Krabbe disease, even if consider that this science may be different from person to person. So some people may have more symptoms than others, above all at the beginning, at the onset of the disease. All these symptoms are explained by the fact that Krabbe disease involves above all central and peripheral nervous system. And here it induces above all demyelination and dysmyelination. Here you can see in central nervous system areas where myelin is absent, so you have the proof of the myelination. Here in this picture, you can see myelin as a blue stain spot. Instead, here you can see here an area typical of neurodegeneration. Instead, here where you can see these larger cells that are stained in violet, you have the appearance of the so-called globoid cells that are macrophages, and I will explain you what are. Here you can see the myelination instead in the peripheral nervous system. Krabbe disease is an inherited genetic disorder. It is an autosomic recessive disorder, so the probability to have a child affected by the disease is for 2 carriers, 25%. The cause of Krabbe disease are mutations in a gene that is called GAL-C. GAL-C gene is mapped onto chromosome 14, and so far more than 130 mutations identified. In Europe, the most common mutation is a large deletion spanning exon 11 to 17. Consider that GAL-C is a very long gene, accounting for 19 exons, and is expressed in above all tissues, but above all in brain and in nervous system. So, which is the protein product of GAL-C? It is an enzyme, a lysosomal enzyme that is called galactosyl cerebrosidase. This protein is synthesized as a precursor in endoplasmic reticulum. Then it moves to the lysosome when it is cleaved into subunits. The subunits combine to form the active enzyme that to perform its activity needs another protein that is called saposin A. Saposins are proteins located in lysosome, which help this type of enzymes. Consider that also some mutations in saposin A gene can have the appearance of some symptoms typical of Krabbe disease. So the mutation related to Krabbe disease in GAL-C gene are loss of function mutation. So it means that these enzymes is not able to perform its enzymatic activity. Normally we see a severe loss, about 90% of enzymatic activity. Krabbe disorder is known also as a lysosomal storage disease because the problem is here in lysosome, so involves lysosomal catabolism. Here you can see the enzymatic activity of galactosyl cerebrosidase. Its typical substrate is galactosyl ceramide, that is a sphingolipid, and this enzyme is able to hydrolyze it into galactose and ceramide. I told you that galactosyl ceramide is a sphingolipid. What are sphingolipids? Sphingolipids are lipids which are constituted by an amino alcohol, that is sphingosine, and it is associated with a fatty acid to give a molecule that is called ceramide. Ceramide is the basic molecule for the synthesis of all sphingolipids. Here you can see sphingomyelin, that is a typical component of all plasma membrane. Here you can see the pathway that led to the synthesis, for example, of gangliosides. Gangliosides are other constituent components of the plasma membranes and are able to interact and to influence the activation of receptors, for example, of epidermal growth factor receptor. And here, from ceramide, you can see the synthesis of galactosyl ceramide and sulfatides. Sulfatides are formed by the addition of a sulfate group to galactosyl ceramide. So, the problem is that galactosyl ceramide and sulfatides are the main lipidic molecules present in myelin. So you understand how a deficiency in their catabolism, in their turnover, can be translated in problems with myelin stability. So you can expect that in Krabbe disease, we can identify an increase in the content of galactosyl ceramide, because I told you that there is a deficiency in the enzyme involved in galactosyl ceramide catabolism. So it is not true. Instead, in the brain of Krabbe disease patient, we can see the increase of another altered substrate, that is psychosin. Psychosin is a galactosyl sphingosine, so a sphingosine with associated residue of galactose. It is also catabolized by GALC, by galactosyl ceramidase, but it is not present in healthy tissue. So it is synthesized by the addition of sphingosine to galactose. And then you can understand that it is not catabolized in Krabbe patients because the enzyme involved in its catabolism is not present, is deficient. But the question is why psychosin is formed in Krabbe disease? Because I told you that in healthy tissues, it is absent. Consider that psychosin was identified at larger amounts in brain of patients after autopsy, just in 1772. But only in recent years, authors demonstrated that another enzyme can transform galactosyl ceramide into psychosin. This enzyme is acidic ceramidase. So you can believe that in Krabbe disease, due to the impairment of galactosyl ceramide catabolism, it is transformed into psychosin, and we see very high levels of psychosin in above all in the central nervous system of Krabbe disease patients. So the problem is that psychosin is very toxic. It is like a detergent. So in particular, it interacts with the plasma membranes, and it alters their organization, their configuration. In particular, the organization of the so-called lipid rafts when signaling pathways start and are organized inside the plasma membrane. So with psychosin, we have an alteration in signaling pathways, several dysfunction in different organelles, like also mitochondria, like lysosome. Briefly, if we treat each type of cells with psychosin, this molecule can induce the death of these cells. And based on the formation of psychosin, you have the explanation of some typical alterations that were recognized in nervous cells of Krabbe disease patients. What about oligodendrocyte? Oligodendrocyte usually express GAL-C and release a lot of cycosine if GAL-C is not active. So cycosine induce the cell death of oligodendrocyte and then the myelination that I told you is the main feature of Krabbe disease. Neurons really do not release cycosine, but it was suggested that cycosine released by oligodendrocyte can induce the death of neurons and so neurodegeneration. Also, macrophages are involved in Krabbe pathology because they try to remove the excess of galactosylceramide and above all cycosine. And in this way, they transform them into larger cells that are called globoid cells. And if you remember, I showed them in the previous picture. The problem is that globoid cells are not able to digest cycosine because they are also GAL-C deficient and acquire a pro-inflammatory phenotype that induces also neuroinflammation. Also, we can see astrocyte activation. So we can you can think that all the pathogenesis of Krabbe disease is clear and that all the biological basis of the pathology are known. So it is not true because recent papers demonstrated that this alteration that I show you are not related in all cases with cycosine levels. This paper was were obtained using animal models of Krabbe disease. This was done with Twitcher mice that were the murine models for Krabbe disease. Instead, this paper was done using zebrafish, in which the galactocerebrosidase knockdown was performed. And above all, in this paper, it was demonstrated that with the GAL-C deficiency, we have an impairment, a severe impairment of brain and nervous system development, but there wasn't accumulation of cycosine. Another important paper is this, in which it was demonstrated that also heterozygote galactocerebrosidase mutants, which are known as carrier, so they are thinking to be perfectly healthy. Really, they also show some dysfunction, in particular in myelin repair. Consider that GAL-C, mutations in GAL-C and also some polymorphism related to GAL-C were also related to the risk to develop another demyelinating disorder, that is multiple sclerosis. So we have the proof that GAL-C activity is important for myelin and stability, despite of the accumulation of cycosine. So in this context, we are performing our research, and we are trying to elucidate the biological mechanism that can be altered in Krabbe disease apart from cycosine accumulation. So for this, we employed above all in this phase of the study fibroblasts that were isolated from patients. Here you will see data collected from 2 healthy fibroblasts and instead 2 Krabbe disease fibroblasts. Why fibroblasts? Because fibroblasts do not produce sarcosine. Instead, you can see here by our mass spectrometry assay that in Krabbe fibroblast, sarcosine is absent as in control fibroblast. So in this condition, we wanted to understand, first of all, if there were alteration in the sphingolipid pattern. Why this? I told you that galactosyl cerebrosidase recognize as substrates galactosyl ceramide and sarcosine, but they are not the unique substrates of these enzymes. Other sphingolipids carry galactose, so can be substrates also for this enzyme. To this type of investigation, we use a technique that is present in my department. In particular, we incubated our fibroblasts with a trishaded precursor for sphingolipid synthesis, that is trishaded sphingosine. We allow to incorporate cells to incorporate sphingosine for about 2 hours, this 2-hour pulse. Then we remove sphingosine and we cultivate cells for other 48 hours. In this time, during this time, we reach the so-called steady state. So we have a balance between the biosynthetic pathways of sphingolipids and also catabolism. Then we extracted sphingolipids and separated them into gangliosides and neutral sphingolipids. Here you can see our results regarding gangliosides. No significant changes were demonstrated. Instead, regarding neutral sphingolipids, we in particular focalized our attention on the increase of lactosyl ceramide in both the 2 fibroblasts affected by C deficiency. Lactosyl ceramide is here. You can see that its level are very low in normal fibroblast, and we have an increase in Krabbe disease fibroblast. So we wanted to understand if this change was related to a problem with the biosynthesis of lactosyl ceramide or with its catabolism. So we performed the previous experiment in another way. In particular, we, in this case, we tried to identify only the effect of the biosynthetic pathway. So we added the sphingosine, trishaded sphingosine for only 2 hours. Also, we added ammonium chloride because it is an inhibitor of lysosomal catabolism. And then we just extracted sphingolipids and perform the same assay that you saw before. Here you can see that lactosyl ceramide is very, very low in almost, it is not detectable in whole cells. And above all, there are no differences among the 4 fibroblasts. Again, we check by real-time PCR the expression of genes related to lactosyl ceramide biosynthesis, and also with its conversion into ganglioside GM3. You can see that any difference has been revealed. Here, maybe for GALT6, you can see an increase in one fibroblast line affected by the disease, but we didn't focalize on this change because the involvement of GALT6 really in the biosynthesis of lactosylceramide is very low. The main enzyme is GALT25. Also, we perform another type of experiment giving to our cells lactosylceramide associated with body P, that is a fluorescent probe. So you can see here that in this probe in Krabbe fibroblast is accumulated in these granular spots that are very low in normal fibroblast. The type of phenotype is typical of lysosomal accumulation of the substrate. So with all these type of experiments, we can conclude that the lactosylceramide catabolism is impaired in Krabbe fibroblast. This can be easily explained because lactosylceramide is a substrate of galactocerebrosidase. Really, also other enzymes can act on this molecule, but we can think that a deficiency of GALC can also produce effect on galactosylceramide levels. And this is important because lactosylceramide is known to be a bioactive single lipid. It means that it is able to regulate some signaling pathway that you can see here, or cellular processes like proliferation, like migration. So many studies on lactosylceramide effect were done on cancer. But what about our cells? In our cells, we try to investigate the main signaling pathway. And we saw that in Krabbe fibroblasts, there was an increase in AKT activation, in PROS4T activation, in BCL2 protein content, in the phosphorylation that is inhibitory of JCK3 beta. So in conclusion, all of these changes are related because PROS4T and GS3 beta are substrate of AKT, and BCL2 expression is regulated by AKT. So we can conclude that in Krabbe fibroblasts, there was an upregulation of AKT. We demonstrated that this alteration is related to lactosylceramide increase. In fact, we incubated healthy fibroblasts with lactosylceramide, and here you can see that you can recognize the increase of AKT phosphorylation. So here we can see our biological model, our molecular mechanism that we identify in absence of cyclosine accumulation, in which we recognize the increment of lactosylceramide, and then the upregulation of AKT and the pathway downstream. So I think you know that AKT is related to the regulation, in particular, of cell survival, of apoptosis resistance. So consider that in particular, most of data regarding AKT upregulation were collected for cancer cells and demonstrated that this is a feature of a pro-survival mechanism. Instead, if a constitutive activation of AKT in, for example, in brain development cannot be so positive because it can impair the process. Consider that in some disorders like autism, there has been an upregulation of AKT was identified. So what about Krabbe disease? We tried to investigate at first cell survival. Here you can see the treatment with different molecules such as etoposide that is able to induce apoptosis or sarcosine itself. But you can see that we haven't changes in survival. Instead, it's most interesting to see what happens with hydrogen peroxide, in particular at this concentration. Hydrogen peroxide was able to induce a major death in Krabbe fibroblasts than in healthy fibroblasts. So this is the contrary of what we think at first, thinking to AKT upregulation. Here you can see the pictures of fibroblasts treated with hydrogen peroxide. We try to understand why Krabbe fibroblasts died with hydrogen peroxide. We think into apoptosis. Here you can see the state of DNA fragmentation. You can see that in Krabbe fibroblasts, there is the absence of DNA fragmentation that is a typical sign of apoptosis. Maybe you can see DNA fragmentation in healthy cells. We think that DNA fragmentation could be a later step of apoptosis. Then we focalized our attention on previous science. And for this, we use JC1 probe, that is a fluorescent probe that can enter into mitochondria. And you can see a different fluorescence emission based on the status of the membrane potential of the mitochondrial membrane potential. In fact, if the potential is correct, is functioning, we have a fluorescence emission in red. Instead, if the mitochondrial potential is broken, so we will see an emission in green. Consider that mitochondrial dysfunction are the first impairment in apoptosis. Here you can see our results. For healthy cells, we have a small but significant increase in green fluorescence after the treatment with hydrogen peroxide. So we can conclude that apoptosis is really induced by hydrogen peroxide in healthy cells. Instead, what about Krabbe disease, Krabbe fibroblast? You can see here that fluorescence in green was not increased. So we can conclude that there are no apoptotic signs after peroxide hydrogen treatment in Krabbe fibroblasts. We also checked the caspase activation, but again, we didn't see any change. So why Krabbe fibroblasts die with hydrogen peroxide? Some work suggested us that BCL2 levels that I remember you are increased in Krabbe fibroblasts can determine a switch from apoptosis to necrosis in the choice of cell die. So we investigated if our cells die through a necrotic process. This was demonstrated for one of our cell line, our Krabbe cell line. In the second, really, at least with lactate dehydrogenase, were not so clear. So now we are investigating in particular regarding this topic, the dysfunction, the lysosomal dysfunction, because it is known that lysosomal dysfunction and the release of a hydrolytic enzyme in the cytosol can induce cell death, also in presence of hydrogen peroxide. So the last thing that I want to show you today is the effect of AKT upregulation of autophagy, because AKT and BCL2 are known to inhibit autophagy. Here you can see our experiment for autophagy that was done inducing autophagy with a serum starvation condition. Also in this experiment, chloroquine was added. Chloroquine is able to block the end of autophagy. So in this way, we are able to check the number of autophagosome, because we just block their formation after the trigger signal, and we block lysosomal degradation. So we see we used a probe, a fluorescent probe again, that is able to mark in particular early autophagosome. And you can see again, that in healthy fibroblast, you can see, I hope, this green autophagosome after 2 hours and 4 hours of treatment. Instead, lower levels of autophagosomes are present in Krabbe fibroblast. Again, here regarding fibroblast, we follow with the activation of LC3B2, and again, which is a marker of autophagosome. Here you can see the experiments done without chloroquine, and here with chloroquine. Again, above all, with chloroquine, you can see that there is an increase in healthy fibroblasts, but not in Krabbe fibroblasts. So what we can conclude with this, with our data. BRC deficiency is related to lactosyl ceramide increase, and this event can alter the activation of signaling pathways, above all AKT. This is related in turn to an altered response to oxidative stress, and also to the capability to trigger to induce autophagy. Consider that both these features are particularly important for neurodegeneration because oxidative stress oxidant molecules are released in a condition of neuroinflammation, for example, by macrophages. And also autophagy is important in this type of condition because with autophagy we can, for example, remove accumulated molecules. So we can have a damage in a further damage for this type of cells. Now, what we are doing, we are trying to transfer this knowledge, in particular on nervous cells, on oligodendrocyte, and to recognize as these altered processes can interfere with the oligodendrocyte survival. This can be important, as I told you, for Krabbe disease, that is our topic, but also possibly to elucidate some mechanism for related to multiple sclerosis. that is another demyelinating disorder, in which there was demonstrated also the involvement of the same enzyme of Krabbe disease. Today, there is no time to show you some preliminary results on this topic, but we demonstrated also the possibility, typical dynamics of GALC of this enzyme also in oligodendrocyte differentiation. So finally, I just a few seconds to thanks all the people who work with me. In particular, a special thanks to Nadia Papini. And thank you very much for your attention. I don't know if there is anybody, questions. Is there anybody?
It's open to question session from participants. If you have any queries, you are most welcome to ask. Any questions from the participants? Once again, I thank you, Professor Dr. Christina. We acquired a lot of information from the techniques and experiments that you have carried out in your research projects. I am very much pleased that you accepted our invitation to this international virtual conference, BICPAC 2022. Thank you, Professor. The day one sessions of this international virtual conference was successfully completed. Tomorrow, the day two will be going to start at 8:30 a.m. So please kindly join with us. Thank you all for your enthusiastic participants in this international virtual conference. Thank you one and all.